Quahog, Los Angeles.
Hey everyone, I'm Shraddha Bonsley. I'm a CMDB major, first year at UCR, and I'm currently living in Austin, Texas.
Good morning, RISE Symposium. My name is Kalei Santiago. I'm an incoming second year majoring in chemistry, and I'm greeting you from sunny San Diego, California.
Hi, this is Leonardo Ballesteros. I am a fourth year chemical engineering student from Fontana.
Hey guys, my name is Brennan Hong. I'm a major of neuroscience. I'm currently a junior at UCR and I'm living in Atlanta right now.
Hello, my name is Morgan Brinkley. I'm currently a fourth year biology major, and I'm from Wildomar, California.
Hello, I'm Raul Raisin. I'm an applied mathematics major, transferring in from Franklin's College, and I am currently in Big Bear Lake, California.
Hello everyone, my name is Jasmine Robledo. I am a physics major, starting my third year at UCR, and I live in Moreno Valley.
Hello everyone, my name is William Troxel. I am a second year biochemistry major, and I live in Moreno Valley.
Hi, my name is Robert Cepeda. I'm a fourth year chemistry major. I'm currently living in Whittier, California.
What up, I'm Judy. I'm a fourth year, and I study inorganic chemistry.
Hi, my name is Ryan Gates. I'm a second year biochemistry major, and I currently live in Riverside.
Hi, my name is Alberto Garcia. I'm an incoming third year mathematics major and currently living in Riverside, California.
Hi, my name is Jasmine Jimenez. I'm currently a third year biology major at UCR, and I live in Upland, California.
Hi, my name is Rachel Paredes. My major is neuroscience, and I'm a sophomore at UC Riverside, and I'm currently living in Fullerton, California.
Hi everyone, my name is Winnet Huang. I'm a third year biology major, and I'm from Rowland Heights, California.
Hi everyone, this is Krishma Uppal. I am a biology major student. I'm transferring to UC Riverside this fall, and I live in Riverside, California.
Hi guys, I'm Ginny Winters. I'm a geology major. I'm in my third year at UCR, and I live in Colvin.
Hey everyone, this is Kevin Watson. I'm a third year biology major, and I currently live in Riverside, California.
Hi, my name is Tuet Nguyen. I am a fourth year bio major here at UCR, and I'm currently living in Riverside.
Hi, my name is Jonathan. I am a sophomore majoring in biology, and I'm from Fremont, California.
Hi, my name is Paul. I'm a fifth year bio major and will be graduating soon. I'm from Thailand, and I'm currently living in Riverside.
All right. So I hope everybody was enjoyed that video. Over the course of the summer, we've had a lot of great students who were extremely engaged with both the research as well as our programming throughout the summer. And we wanted to have a few of our students speak to you and share more about their introduction. So they'll tell you a little bit more about themselves and what RISE has meant for them. So our very first student speaker of this morning is going to be Mr. Ernesto Gonzalez. Ernesto, feel free, take it away.
Excuse me. Good morning, everyone. My name is Ernesto Gonzalez, and yes, today I'll be talking today about my experience in RISE. Again, I'm a second year student majoring in neuroscience. I'm also a first generation student. And I just want to talk about how RISE has broadened my perspective in regards to the diversity of research topics and research themes, because it can be really interesting at times to consider that the precise and focused nature of research can sort of create a tunnel vision, especially now in remote working environments. This cultivates a sort of isolating experience that may drive students to reconsider or adapt their career choice. And as minority students, whether that be as an ethnic minority, a religious minority, or even our LGBTQ students, it can be really daunting to consider life in a competitive field like research. But my experience in RISE has quelled those concerns because from day one, RISE has established a sense of community among different programs, whether that be the CNAS LC scholars in the B2B program, among different disciplines, whether that be myself in neuroscience or my peers in chemistry and biochemistry, or even different students, because even in the small friend group that I've made, none of us look like each other. And I've had the pleasure, again, engaging with like-minded peers in a really welcoming and nurturing environment. that has really only further encouraged my passion for research. I feel that I've developed a really strong connection with individuals who I may have never met otherwise, like Dr. Connie Nugent. And so RISE, again, has generously provided in regards to professional development as well. In any career, it is often the first step that poses the greatest challenge. And in my experience as a mentor to my peers, to those in my residence hall, or even just to my friends, many students often approach research with trepidation, sort of stumbling around in the darkness. And this experience is really confusing, usually because there's a lot of uncertainty as to what a career in academia might look like for them, right? Whether that be working as a professor or working in industry. And even students who have developed a really concrete idea of where they want to go, there's still plenty of puzzlement as to, okay, I know where I want to go, but how do I get there? Just because there's so many paths to take in research, whether that be taking a master's or pursuing a PhD. But RISE sort of provides as this penultimate opportunity in that students are provided with a chance to get some real experience, right? That first research experience. And not only that, there's also a chance for exposure to a variety of different subjects and projects, right? Alluding to what I had mentioned earlier. And for some students, myself included, this program offered a platform to demonstrate our skills and our commitment to our mentors in our lab, sort of a stage to prove ourselves. Because working collaboratively on a project with a member of the lab is often an opportunity that may have otherwise only been possible in the 3rd or 4th year, right? Because as a part of RISE, we are working one-on-one, whether it be with a graduate student or a postdoc student, or even the PI themselves, to work and see this project to its end. And so even in my small group, for instance, we've actually begun talking about like potential interdisciplinary projects, whether that be me collaborating with the biochemistry department or me collaborating with the geology department. And so just to wrap things up, RISE has really been a fulfilling experience for myself and my peers. And I'm sure we're all feeling a little bittersweet sense of accomplishment here at the end.
All right. Thank you very much, Ernesto. Great job. And now we'll have our next speaker, Ms. Wineth Wong.
Hi, good morning, everybody. My name is Wineth Wong, and I'm a 3rd year biology major. I'm currently part of the PERSIST branch of RISE. So PERSIST is a sophomore mentoring program that a cohort of 12 students go through. They are mentored by staff, faculty, and peers before joining RISE for the summer research program. When I was looking into RISE and PERSIST, I was just looking for a way to broaden my horizons. My first research lab experience wasn't that great. I was just not getting the experience that I wanted. So it was probably due to the miscommunication and not knowing how to advocate for myself. And this led me to apply for RISE, and lucky for me, I got in. And one of the best parts of being a part of RISE and PERSIST is the monthly meetings, the brown bag meetings with Dr. Nugent and Dr. Wessler, and creating a close relationship with these people and receiving a lot of great academic advice as well as professional advice. And some of this advice includes graduate school, publications, and even the small things like how to read an academic paper. Fortunately enough, I was able to become close enough with Dr. Nugent to have her listed as one of the job references that I currently have now for the next academic year. And I want to tell you a little bit more about my current research experience in a different lab. I'm currently a part of Dr. Anand Sankaray's Molecular Cell and Systems Biology lab. I have been a part of his lab since the winter quarter of my 2nd year. That's around January of this 2020 year. And when I first came on, I was actually working on a really cool project funded by D, where we got to mutate the genes of flies using CRISPR-Cas9. And so I was able to study the life cycle of a fly, and I was able to collect eggs as well as inject some of these eggs with CRISPR-Cas9 and then see if they would grow up and survive to become mutant flies. unfortunately, due to COVID and no more funding of that project, I'm working on a bioinformatics project that I'll be presenting about a bit later today. And so not only has RISE shaped my research experience, but it's also shown me what being a STEM major has to offer. Because when I came into UCR, a lot of my peers were on the pre-med track, and that was pretty much the only thing that I had thought about too. I'm super guilty of that. I'm a pre-PA student, and I just wanted to see if there were other options out there for me professionally. So because I've been going to all these RISE and Persist workshops and seminars, I've actually been able to meet a lot of different professionals who do a lot of different jobs from all different types of STEM majors. And so that it actually encouraged me to reach out to a friend's mother, because she was actually a clinical lab assistant at USC for a little bit, working in their oncology department. And I thought, how cool is it that she gets to do medical research? So that further encouraged me to look into what being a physician's assistant has to offer me. And lucky enough, being a physician's assistant, I can also participate in research, and specifically the medical research like my friend's mother did at USC. Because of RISE and Persist, I've had so many opportunities, and I was able to grow as a person, academically and professionally. And now I work in a lab that I have such supporting staff in, and I have such passion for the project that we work on. And I am able to meet so many friends in my cohort of Persist and in my lab, and RISE is such a great opportunity to just see what research has to offer academically and professionally. Thank you so much for listening to me talk. Please enjoy the rest of the presentation. Have a good day.
Thank you very much, Gwyneth. And last but not least, we have our third speaker of the morning, Ms. Jenny Winters.
Hi, good morning, everyone. I'm Jenny Winters. I'm a transfer student from Riverside Community College. I'll be starting my third year at UCR this October, and I'm so excited to be pursuing a bachelor's degree in general geology. The RISE program offered me the opportunity to experience research on a more personal level and even explore a subject entirely new. Under Dr. Purcell's 6-leg program, I was able to work with Dr. Reddick in the entomology department. I was able to form a hypothesis, create a protocol that I could replicate, and even got to collect my own data. As in many researching projects, I faced a challenge at this point. Collecting data can be one of the most rewarding parts of an observation. However, it could be also one of the most frustrating ones. My project involved observing ants in their natural environment for about 2 weeks. At times, I was able to locate my species, and at other times, I wasn't. Sometimes workers that I did see were so disorganized that I didn't even know how to categorize their behavior. This challenge of not having the data that I envisioned grew on me. But with only 8 weeks to conduct an observation, I didn't have time to make any adjustments, so I had to run with the data that I had. This portion of the scientific method showed that as a researcher, we are subjected to discrepancies, and it is part of our job to develop alternate methods on how we can work around those challenges and hopefully extract more focused data. Thankfully, I was able to recognize this through a hands-on approach, giving me a true in-depth feeling of what research is. The process was difficult, but it was rewarding. You are faced with puzzling questions in which you strive to answer by examining and understanding your data in hopes that you discover something entirely much larger. As a geology major, I am inspired to incorporate research as a main component in my career and go beyond what I thought was my comfort level and accomplish much more. Outside of my project, because we did have some scheduled fun time, I was able to network, bond, and surround myself with like-minded individuals. Although in a virtual setting, we are still able to share ideas, ask questions, and motivate each other. Our peer mentors and the RISE coordinators were there every step of the way to ensure we had all the resources available to accomplish our tasks. The peer mentors also created fantastic social nights that allowed us to connect, and we even got to test the virtual capabilities of Zoom. For the record, virtual Pictionary was one of my favorites. And I found this important because research also involves the partnerships that we build. And who knows, perhaps one day we may get the chance to see another together and collaborate on a project while we share a fond memory of our RISE experience. When asked to share a thought to my peers and future RISE participants, I had to think for a minute, which did turn into a day or two, but I got it. No task is too great or beyond your capabilities. Seeing the RISE application in my email, I was hesitant. As a transfer student, I already had so much on my mind before fall, but the 8 weeks seemed like a good distraction. So please, take this little piece of advice. In an office hour, I was told that something that completely encompassed these past 8 weeks. Science is an interdisciplinary subject, and expanding our knowledge can only further our ability to answer the question why. And while that may make you more curious, that's okay. It should push you to look closer. So please, do not hold back. Let your mind wander. Explore as much or as little as you want. And finally, make opportunities happen by going after them. Thank you.
Thank you very much, Ginny, for your very inspiring talk. So now we go on to the presentation portion, and we will start off with our oral presentations. Our very first group comes from Dr. Dawn Nagel's research experience for undergraduates of sorts. She had overseen a group of students over the summer, and this is actually a group presentation for our orals. So we will have them present, and they will take it over from here. Thank you very much.
Well, hello guys, I'm part of Dr. Nagel's lab, as well as Carolina Acevedo, Kiani Kosek, Andres Perales, and I'm Eric Torres. Thank you for the introduction. I will be introducing what my group worked on this summer. Each of us will be presenting the different aspects of this research, and over the summer we had the opportunity to work with Dr. Nagel's lab in trying to understand how genes are regulated by the circadian clock and how they respond to heat stress, particularly rice. Next slide, please. Now, as we've seen, there has been and will continue to be an increase in the world's population, so the demand for an increase in crop production will also continue to grow. Climate change has affected crop productivity around the world, and one of the most affected crops, as well as simultaneously one of the most highly produced crops, happens to be rice. And as mentioned before, climate change will continue to affect these crops, and one of the ways it will continue to affect these crops is by inducing their growth and development, which would ultimately lead to quality and yield reduction in rice. Next slide, please. One of the ways that plants deal with changes to their environment is through the circadian clock. What the circadian clock does is that it integrates signals from their environment and regulates all of these developmental processes, as you've seen here. Next slide, please. The Nagel lab was interested in understanding how each stress really impacts the gene in Arabidopsis, or plants in general, and how the time of day, circadian clock, and/or heat stress impacts genes in the genome. What was done to check to see how the genes are responding to heat stress is apply heat stress at different times of the day, once applied in the morning and once applied in the afternoon. What the Nagel lab found was a set of genes that shows a difference depending on the time of day that heat stress was applied. From this data set, 4 genes were selected for further study for my group. Next slide, please. These are the 4 genes that were involved in this project. In addition to being involved with abiotic stress responses, these genes were also involved in some kind of growth or developmental responses in Arabidopsis. So these are the 4 genes that we found in Arabidopsis for further study, as Kia will continue to explain.
Next slide, please. Thank you, Eric. To visualize what that RNA sequence experiment produced that Eric mentioned earlier, we referenced its data best based on Arabidopsis, and that showed us the mRNA expression of our genes in response to heat stress at 2 different times of the day. For the first gene, there was a response at both time frames, ZT1 and ZT6. That ZT is relative to the time when light was first applied to the plant, so those numbers 1 and 6 show how long the light has been on. I also want to point out that for this gene, there was a significant decrease in the afternoon compared to during the morning. As for the other genes, they only showed 1 response to 1 time frame. Next slide, please. Another area that we wanted to examine to support our selection for these 4 genes was the circadian expression of Arabidopsis. This data was sourced from a public journal database that has data for genes that are regulated by the circadian clock. And overall, we found that our genes showed a cyclic pattern with peaks at particular times of the day. Next slide, please. Moving forward with our research, our main goal was to identify the orthologs of our 4 Arabidopsis heat response genes and to see if we could identify them also in rice. And from there, we wanted to determine whether the circadian expression was conserved across the species. That was the experimental approach. We also used bioinformatics to determine whether the expression of both our Arabidopsis and our rice genes would have a change in response to other abiotic stresses, such as cold and drought. Next slide, please. And now let's dive into our experimental approach. For week 1 through 5, the agenda was to identify our rice gene of interest, amplify it, clone it into bacteria, check the sequence to make sure it's there and actually correct, and then clone it again into a vector. For week 6, we performed a tobacco infiltration, and we used the same vector as we had cloned the information in, and we used an enzyme called luciferase to make sure that we can monitor the expression of our genes within the plant. For week 7, we analyzed the rhythmic response and compared it to the journal data of rice. And I will hand it off to Andreas to elaborate more on that topic.
Thank you, Kio. So then we first started by identifying our genes in rice. We used the Rice Genome Project using a blast search of our amino acid sequence for Arabidopsis to find our rice homologs. We then annotated our rice genes and designed forward and reverse primers for our promoter regions, which will lead us into our next slide. We then used these primers to form a PCR reaction, and then once our promoters were amplified, we then ran an agarose gel to ensure that our PCR reaction was successful. We then cloned our promoters into our desired plasmid and plated it, after which we let colonies grow on our plate and formed a colony PCR, and which allowed us to see that our promoter was inside of our plasmid. And then we performed a sequence analysis to ensure that our promoter was inserted into our plasmid correctly. Next slide, please. We then transformed our plasmid into Agrobacteria using electroporation, which using an electric field to pull our plasmids into our bacteria. And then bottom left, you can see there's an electroporator machine. And on the bottom right, we then plated our bacteria and then did swabs of our colonies and had it spit an infiltration buffer, which leads us into our next slide. We then infiltrated our tobacco leaves. As you can see in the second picture, the leaves appear to have some wet areas. That's because they were injected with a needleless syringe of our bacteria that was inside of our infiltration buffer. We then have them imaged for every hour for 4 days. And to the right, you can see these are the results, and they're glowing because that's the luciferase being expressed. We then decide to analyze these images, which is going to be on the next slide. And I'll leave it to Carolina to explain from here.
Thank you, Andres. So as Andres previously mentioned, once we successfully infiltrate the tobacco leaves, we can then image them at constant light for 4 days and have an image taken at 1 hour increments. We are then able to gather and process imaging data from a publicly available and commonly used software called ImageJ. The expression level values of our genes of interest in the tobacco leaves were processed using the software. As you may have already noticed, our 2 graphs at the top only display 2 genes of interest. The imaging data results displayed here are 2 genes we were able to get thus far with. The other 2 incomplete genes of interest will be processed and completed at a later time. Once we have gathered our imaging data, we will then put those values into Excel. Once our data was plotted, we examined the circadian expression profile of these 2 genes. What you can see on the 2 graphs above, which may not be obvious, is that there are peak maximums and peak minimums. This rhythmic profile exemplifies oscillations, which can also be compared and similar to the rice circadian expression graph below. Next slide, please. So as Kiani mentioned earlier, we had two main goals in our project. Goal number 2 was a bioinformatic approach, meaning the next thing we wanted to do was look to see whether these genes are responsive to other types of abiotic stresses. For our project this summer, our main focus was cold and drought abiotic stresses. So we surveyed and gathered data from another publicly available database called ePlant. We examined the expression levels of these two abiotic stresses in both the shoot and root of Arabidopsis and rice. Next slide, please. So in Arabidopsis, both the root and shoot tissue, we noticed that it responded to cold. And in some cases of our genes, they also responded to drought. In the bottom graph, you can also see that in general, rice was observed to be responsive to both the drought and cold abiotic stresses. So these values suggest that these genes are actually pretty good targets to be dissected further. Next slide, please. So overall, what we found was that the timing of expression and response to abiotic stress differs for most of the genes between the two species. One of the next steps for this project is to examine whether or not these rice genes respond to heat stress. Another next step of this project is to finish cloning the 2 remaining rice genes that were incomplete in our project. So what we would do is clone them, and then we will insert them into a p-enter vector and then store them later for further use. So lastly, our long-term goal of this project is to be able to use the information we have learned to create rice lines that will likely be more tolerant to heat stress. Next slide, please. So we would like to thank our wonderful Dr. Nagel, our PI. We'd like to thank her Nagel Lab team, and we'd also like to thank our RISE coordinators, our peer mentors, and most of all, the project funding from the National Science Foundation. Thank you. We would love to take your questions if you have any right now.
All right, thank you very much. So for all the guests, oral presentations are 10 minutes with 2 minutes of questions. If you would like to ask any questions, please type it in the chat, or you can raise your hand and we can unmute you and you can ask it live if you'd like. And I do second Lauren's great presentation, really great job. And you guys had every single slide went really smoothly. Okay. Are there any questions for our panelists today? All right, well, we were running a little short, so we're actually gonna go ahead and move on to our next presenter. So Ms. Kitty Lamb, actually for Dr. Hyam's lab, would you mind, she will go ahead and unmute. And we will go ahead and start the next slide. Thank you very much.
Hi, everyone. My name is Kitty Lamb, and I am a third-year biology student in the PERSIST program. Throughout RISE, I have been working with my faculty mentor, Dr. Tim Hyam, on the research topic of gecko locomotion. Specifically, we are looking at how tail autonomy impacts the jumping performance of crested geckos. And in my next slide, I will introduce these topics. So what is autonomy? It is the voluntary loss or self-amputation of an appendage. Many animals, such as spiders, crabs, and starfish, exhibit this behavior. However, the most common type is tail autonomy, which is most widely found in lizards. The mechanism that goes into tail autonomy includes specialized muscles, spinal cords, and vertebrae that allow the lizard to completely remove its tail. This behavior is often provoked when the lizards are threatened by predators, and geckos in particular have the ability to fling their tails away from their body, which will keep moving and twitching to distract the predator, allowing the gecko to escape. Another unique trait that geckos have is a characteristic jumping posture. As you can see in the picture below, geckos tend to outstretch their arms forward and their hind limbs backwards as they jump. They have also been studied to be well adapted to changes in body position as they jump, and an important factor that contributes to this is their tails. So although tail autonomy helps geckos to escape predators, it might actually cause harm to their jumping performance. For instance, previous studies involving anolis lizards have shown that tail autonomy significantly impacts their in-air stability when they jump. With this in mind, we wanted to see if tail autonomy would affect geckos in a similar way. But first, I will give background on the particular gecko species we have been studying. We are studying the crested geckos. They are native to the islands of New Caledonia off the coast of Australia. They are an arboreal species, meaning that they live in the trees and are mostly found up in the canopy. Interestingly, these geckos were thought to be extinct until they were rediscovered by a group of scientists in 1994. Because of their recent rediscovery, there is not much literature or studies that focus on their locomotion or tail autonomy, so this research is one of the first of its kind. Another reason why we are looking at crested geckos to study tail autonomy is the fact that, unlike most species of gecko, crested geckos cannot grow their tails back after it has been lost. Because of this, we wanted to see how tail autonomy would affect these geckos differently compared to other geckos, which leads me to my research question in the next slide. The research question that we have been developing this summer is, how does tail autonomy impact the jumping performance of crested geckos? As I mentioned earlier, geckos have a specific jumping posture and great jumping adaptability, so this research will help us to understand how permanent tail loss would affect their jumping performance. And in my next slide, I'll describe the methods and materials. For this project, I have been digitizing and analyzing high-speed videos of geckos jumping, as shown on the right side. These videos were collected by a professor from New Caledonia using high-speed cameras that recorded at 400 frames per second. Each gecko was recorded jumping, pre-autonomy, and post-autonomy, and how tail autonomy was induced in the geckos was by gently squeezing their tails at the base until the gecko released their tails voluntarily. With the high-speed videos, I digitized them with a software called MATLAB and an app called DLT DV8. In the bottom right corner, I included a screenshot of what the software looks like. With MATLAB and DLT DV8, I was able to go through each frame of the high-speed video and track where certain points are located as the gecko jumped. The points I digitized were the snout, the hind limb, and the lowest part of their body, which are important for studying how the velocity, propulsion, and the landing of the jump might have been affected by tail loss. However, due to time constraints, we are only able to analyze how the velocity of the jumps are affected by tail autonomy. So in order to find velocity, I used a program called RStudio. I input the X and Y coordinates that I got from digitizing into an equation to get velocity and graphed it. Because there may have been inconsistencies in the data, RStudio also allows me to filter the data to take out the points that were either too high or too low compared to what I collected overall. This gives me a nicer, cleaner looking graph, which I'll show in my next slide. The first graph on the left shows the velocities of the jump pre-autonomy, and the jump in the second graph on the right shows the velocities of the jumps post-autonomy for the same gecko. These graphs show all the velocity points that were digitized from the snout throughout the entire jump. To break down what you see here, the beginning of the graphs is where the geckos are quickly raising their snouts as they prepare to jump. The middle is where the gecko jumps and takes off into the air, and the highest peaks near the end indicates where the gecko has landed because their snouts move the fastest as the body impacts on the surface. The part I focused on in the graph is where the gecko is taking off. This is because that's where the actual jumping occurs. So taking those points near the middle across all gecko videos, I was able to average the velocities of each gecko pre-autonomy and post-autonomy and put them into a bar graph, which I'll show in my next slide. The bar graph compares the average velocities of all jumps. It shows that on average, the velocities of jumps pre-autonomy was around 0.25 centimeters per second, and the average velocities of jumps post-autonomy was around 0.2 centimeters per second. And in my next slide, I will discuss what these results imply. From the bar graphs before, it appears that crested geckos pre-autonomy had higher average velocity jumps than during post-autonomy. This could indicate that tail autonomy hinders or lowers the jumping performance of crested geckos. However, the difference between the two velocities are relatively small at around 0.05 centimeters per second. With such a negligible difference in average velocities, it could actually indicate that crested geckos are perfectly adapted to tail loss. This makes sense because they are unable to grow their tail back, so they must have evolved to adapt really well to losing their tails. And that's super fascinating because it differs from the previous studies that I mentioned earlier. However, future research is needed to examine this, which I will discuss in my next slide. And so my next steps in this project would be to continue analyzing the data to understand how propulsion and landing are affected after autonomy. I would then conduct statistical analysis on the results to prove whether tail autonomy does or does not have a significant impact on jumping performance of crested geckos. Another topic that could be researched in the future would be how tail autonomy impacts other gecko behaviors, such as acquiring mates or food, as it hasn't been extensively studied yet. Besides that, studying tail autonomy could have greater future implications for spinal cord injuries in humans, since lizards have the ability to sever their spinal cords with minimal to no damage to their body. Studying how they are able to adapt to losing parts of their spines could pave the way for helping humans with understanding and possibly treating spinal cord injuries or trauma. That wraps up my presentation. So in my next slides are my references and acknowledgements. I would like to give a huge thank you to my faculty mentor, Dr. Tim Hyam, grad student Marina Volin, my fellow researcher, Maya Rodriguez. I want to say thank you to my amazing peer mentor, Gina Zoe, the people of Persist and Rise, Alex Cortez, Mi Tran, Marissa Carranza, Dr. Connie Nugent, Dr. Sue Wessler, Jen Copia, Jaren Patchke. I would also like to thank the organizers of Persist, Rise, and CNAS Scholars for this amazing opportunity and support. Lastly, I would like to thank everyone for attending and listening to my presentation. I will gladly answer any questions or comments. Thank you.
Okay, great job, Kitty. If anybody would like to type any questions, we'll give you a moment to write that up. Or if you would like to ask live, you can raise your hand and we can unmute you to ask your question directly. I did have a question for you, Kitty. What was the most interesting part of working with working not just with the data, but with reptiles and this lab for the summer?
Good question, Lee. I think the most interesting thing about this lab was just learning how adaptive geckos were and their specific body parts that they evolved to be just amazing at being agile. And how they're just amazing creatures. And overall, I think just using the programs, they're very new to me, so it was very interesting to, do trial and error and just learning how to work them.
All right. Looks like you actually have a couple questions from Ernesto. He has asked, has your lab considered estimation tools like Deep Lab Cut for behavior video analysis?
I don't believe so, but I can probably ask for you.
And you have another question from Fatima. What was the most difficult part when analyzing the high-speed videos?
That's a good question. So I think one of the most difficult parts of the high-speed video or analyzing them was just trying to find the parts of the gecko, because like when they're like slowed down like that, it's actually like some parts get blurry when I'm digitizing them. So some parts I'm like, is that a knee, or like is that like their body? So yeah, it's just the blurriness of it is just one of the most difficult parts, but I found it really interesting and fun.
All right, thank you very much. So since you do happen to have a couple more questions in our Q&A, we'll actually keep you on the panelists, and Kitty, you can actually type in the answers and and once you type it in, it'll be displayed for everybody. So in the interest of time, we're going to be moving on to our next speaker, but we will also keep these questions so everyone can see on the Q&A, and then you can feel free to try to answer them.
Thank you.
All right, thank you very much. So on to our next presenter, Ms. Maya Rodriguez, and I will have her we will actually activate her video, and her slide is up. So Maya, please take it away.
Hello, everyone. My name is Maya Rodriguez, and I am a third-year biology major transfer student from Chaffee Community College, and I am a part of the Summer to Bridge research program. I will be presenting to you today about the effects of incline on locomotion in an arboreal gecko. Locomotion is the movement to go from one point to another, and this ability to do so is incredibly useful for animals in tasks like capturing prey, evading predators, and even interacting with each other. And by conducting all of these tasks, it is likely done at different inclines. So how, if at all, does incline affect locomotion? Well, when talking about humans, our locomotion is definitely impacted by incline, due to the fact that we typically spend our time on a level plane, and because of this, our bodies are accustomed to handle such environments. However, if we were to try and, say, climb a mountain, for example, with many steep inclines, it would take us much longer to walk a mile up a mountain than to walk a mile in a level park. And some animals face many incline changes in their daily environment, so over time, they have developed certain features to help increase their locomotive performance. For example, mountain goats, which you can see here, travel on incredibly steep slopes, and because of this, they have adapted cloven hooves with 2 toes that spread wide to improve balance and have rough pads on the bottoms of each toe to improve grip, similar to a hiker's boot. Along with spiders who have thousands of tiny hairs at the ends of their legs, which creates multiple contact points between the spider and the surface it is on, which allows it to hang onto all types of surface angles. And today I will be presenting on how inclines affect the locomotor performance of Gonatodes ceciliae. Now, this type of gecko genus has not been widely explored in the research world, which is particularly due to the fact that this type of genus does not have an adhesive abilities, except for one species, which is showing beginning signs of adhesion. Typically, geckos with adhesive mechanisms are the most studied and researched by scientists, which have led to many technical advancements, such as better adhesive tape, improvements in traction and grip on shoes, and better elastic adhesive bandage material. So who knows what incredible advancements in technology can come out of this new groundbreaking research on this particular gecko genus. Here's a little bit about Gonatodes ceciliae. That it is found in the northern region of South America, particularly Trinidad, which is where the geckos from this research project is from, and Venezuela. They are arboreal animals, meaning that they live in trees, and a bit about their description is that this particular type of male gecko can grow as long as 12 to 14 centimeters, and it has a brown base color with bright yellow and red specks throughout their body, as you can see here. Whereas the females can grow as long as 11 centimeters and have a beige base with dark brown patterns. The methods and materials that were used to conduct this research experiment was a high-speed camera that was used to capture the geckos moving up the inclined platform, as you can see here in this video. The running platform that was used was lined with sandpaper in order to try and resemble the gecko's natural habitat's texture. This platform was tilted at 0, 30, and 90-degree angles to see how incline would affect its locomotion. Then the video would be uploaded to a software program called MATLAB, to which I used an app in the program called DLT-DV8, which was then used to digitize different points on the gecko's body, such as its snout, hind limb, and hind foot. In this picture below the video, you can see a screenshot of what the program looks like when digitizing. The pink line in the program is tracking the nose in this particular example. Then after digitizing, it would create an Excel sheet of all of the points digitized, which would then be uploaded to another software program called RStudio. And on RStudio, I coded the program to calculate the velocity of all of the points, which produced another Excel sheet and a graph, as you can see on the bottom left corner. This is an example of what the graphs that were produced by RStudio looked like for a particular gecko running at 0, 30, and 90-degree inclines. For these graphs, you can see the maximum velocity of the gecko during its run at a 0-degree incline was about 5.5 pixels per second. At a 30-degree incline, the maximum was just below 4 pixels per second. And at a 90-degree incline, its maximum velocity during its run was about 5 pixels per second. From this data, you can infer for this individual that it traveled at its highest velocity at a 0-degree incline and lowest at a 30-degree incline. 2 other geckos were also tested at these different inclines, and with all of their average highest velocities during their strides while running, the following bar graph was created. So for this bar graph, the y-axis units were changed from pixels per second, like in the last graphs, and converted to centimeters per second, and this was done to make the y units more relative to the velocity metric system, units which are in centimeters per second. This bar graph focuses and is composed of all of the 3 geckos' highest velocity averages during their running strides as they ran up the different inclines 0, 30, and 90 degrees. Whereas the RStudio graphs were in the last slide were composed of the velocity at the tip of the snout. And the result outcomes were the same for both, being that the highest velocity during a running stride was taken at a 0-degree incline, the second highest being the 90-degree incline, and the third highest being a 30-degree incline. These results are very interesting since one would think that the hardest incline to run up would be the 90-degree incline. However, this is not the case. Some theories as to why this could be is due to the possibility that 0 and 90-degree inclines are more common in their natural environment, which are trees. The next steps would be to do the exact same trials and test runs at the same inclines with the other species of geckos, Vitatus, Ocellatus, and Humoralis. After we conduct these steps, we can focus on comparing Gonatodes humoralis, which is the species that has the beginning signs of adhesion, with the other 3 gecko species to see if its beginning development of an adhesive system help or hinder the gecko's locomotive performance when facing diverse inclines. Other research studies have shown that an adhesive system does in fact hinder velocity, so it'll be interesting to see if it is still applicable to this case and at different inclines. This concludes the research portion of my presentation, and the following are my references and acknowledgements. I would like to thank my faculty mentor, Dr. Timothy Hyam, my fellow researcher, Kitty Lamb, graduate student Marissa Bolin, my peer mentors, Gina and David, and the following organizers and the primary organizers of RISE, who are Nhi Tran and Marissa Carranza, along with all of the CNAS alumni and RISE donors, in particular, Mr. Kenneth and Nancy Gray, who funded my participation in the RISE program. Thank you so much for this amazing opportunity. I will now be open for questions.
All right, thank you very much, Maya. If anybody has any questions, again, feel free, please type into the chat, or you could even try the Q&A. It looks like Bailey did ask a question. What was the most challenging obstacle you encountered throughout the duration of your research? And amazing job.
I would say the most challenging part would be learning all of the, how to work all of the software programs, just because I did not particularly know much about computers in general before this research program. So I definitely received an amazing crash course on how to work my computer.
All right. Any other questions? I think we have another, just in case anybody needs time to type up here. And again, you're getting plenty of compliments for all panelists. Great job. All right, and it looks like Radhika did have one question. Did you control any other factors in the trials to mimic the natural environment of the animal?
No, I don't believe so. I particularly wasn't there when the videos were being shot and when the trials were going on. But as far as I know, the sandpaper was the key element in resembling its natural habitat's texture.
All right. Well, thank you very much. We are actually at the end of your presentation. Thank you for keeping on time and very great presentation. We will move on to our next presenter, Mr. Ernesto Gonzalez, whom we saw earlier today. So thank you very much, Ernesto. You are ready to go.
Good morning, everyone. My name is Ernesto Gonzalez, and today I'm going to be talking to you about my work in the mechanisms underlying social behavior deficits after traumatic brain injury. So on the next slide, we'll get started with a bit of background on the hippocampus, which is pictured in the top right, but more specifically, we focus on a subregion called the CA2, which is pictured in red. And we know the hippocampus is a limbic structure found in humans and rodents and is essential for learning and memory. Interestingly enough, human patients who have suffered a traumatic brain injury or TBI often report damage to the hippocampus and social behavior deficits. This is interesting when taken in conjunction to a study in which CA2 neurons were silenced in rodents, and the results were that we saw impairments in social memory, but not in sociability. This indicates that CA2 is in fact critical for social memory. And so I want to understand more about this possible connection between CA2 and TBI, traumatic brain injury. And in order to do that, we're using the fluid percussion injury model, which is pictured in the bottom right. And this is just a means of creating a diffuse injury in the, of course, anesthetized mouse brain. On the next slide, we can talk about the objectives I set out to complete in the duration of RISE. And first, I wanted to assess the social behavior deficits in mice following brain injury. And two, I wanted to quantify the magnitude of cell loss after injury. And so if we look in the bottom right, we can see another view at CA2. This is a blue and green image, but really it's the bright green you want to focus on. The bright green there is CA2. And this was done using STEP, which is a marker for CA2. And on the next slide, we can talk about my first aim, which was to assess social behavior deficits. And so 1 week after injury, mice were tested for sociability and social novelty preference. And so next slide. You can see here that in our 3-chamber maze, we use this for both tasks. In both tasks, the mouse starts in the center. And in sociability, we put a new mouse, stranger 1, in the top cup, and we leave the bottom cup empty. And this is to test whether the mouse will interact more with the novel mouse with an object. And the second task, social novelty preference, we leave stranger 1 up here in the top cup. The mouse, again, starts in the center chamber, but now we have a new mouse, stranger 2, in the bottom cup. And so in social novelty preference, we want to see, will the mouse interact more with the novel mouse or the familiar mouse, or stranger 2 or stranger 1? This is really testing, this is a measure for social recognition memory, because the mouse, our test mouse, should be able to remember that they've already interacted with stranger 1, and so stranger 1 is a familiar mouse. But if CA2 is damaged, and as we know, CA2 is critical for social recognition memory, they won't be able to make that sort of distinction. And so as you can see, these behavior tasks are recorded as videos, and we use a software called DeepLabCut to analyze these videos. And first, DeepLabCut extracts all the frames because a video is just a set of frames, right? And so for a 10-minute behavior task, that looks like around 18,000 frames. And so DeepLabCut will give me, the user, 18 to 20 frames and ask me to label them. And as you can see, I've chosen to label the head blue, the center green, and the base of the tail red. And so again, I'll label those 18 to 20 frames, and then DeepLabCut goes ahead and labels the other 18,000 frames. And those frames are then put together, and you see a nice labeled video like this. The video looks nice, but more importantly, we also get coordinate files. So spreadsheets full of coordinates giving us the exact location of the head, center, and tail of the mouse for the entire duration of the video. And so on the next slide, we'll see how we can take those coordinate files and get social interaction quantifications. If you look on the left side here, we have, social interactions in mice have been previously defined as a point in time in which the nose or snout of the mouse is within 2 centimeters of another cup. And so that's plotted here with white circles using matplotlib. And so if we take these plots, right, we've defined these social interaction zones. If we take these zones in combination with the data we got from DeepLabCut, we get the plot you see on the right, where each of these blue dots represents the single frame of the video and where the snout was for that single frame. And so we can take these together, and let's say, for instance, that the camera is recording at 30 frames per second, and I have 30 frames where the snout was within the zone for cup A, then I can therefore say that the mouse interacted with cup A for 1 second, right? And so on the next slide, we can see that I can take these durations and calculate the discrimination index. And so the discrimination index is a measure of comparing how much time was spent with one cup versus the other cup. So in sociability, that's stranger 1 versus an empty cup. And in social novelty preference, that's stranger 2 versus stranger 1. And that produces these figures you see on the right. But again, this is just preliminary data. But we went ahead anyway, you can see the error bars are pretty large. We went ahead anyways and plugged it into a SciPy function for the Shapiro locomotion test. And this test failed, which indicated that the data were normally distributed. So we also went ahead and did a student's t-test and found that the data were not statistically significant yet. But again, this is just preliminary data. We use a very small sample size, 5 injured, 4 uninjured. And traditionally in behavior, a sample size of 10 or more is acceptable. But again, that's our future work. And so on the next slide, we can see more applications of DeepLabCut. And so you see we have a new video with a lot more labels. So I mentioned earlier that we are quantifying social interaction zones, right? Those white circles that we talked about earlier. So it's really important that we're really accurate in terms of where those zones are, because we're using those zones to quantify social interactions, right? And so the 3 labels you see here around each of the cups, so in the top cup, that's those blue and green labels, we're using those labels to triangulate the precise center of the cup. And that's using this equation you see here at the bottom of H and K. And we did this for both cups, and that gives us a more accurate representation of those social interaction zones. But we also have these new corner labels, these orange and yellow labels, and those allow us with the Pythagorean theorem to create right triangles that then create boxes that more importantly give us a representation of the chambers. Because let's say, for instance, that the mouse isn't interacting with cup A, the top cup, but maybe it's just hanging around near the cup. We might see that if we start looking at time spent in each chamber. And so if you look on the right, we have a new plot, again, using these new labels, that shows us very specific, very precise depictions of the chambers and of the cups. And this would be used in conjunction with the DeepLabCut data as we saw previously. But that wraps up what we've done with the first aim, quantifying behavior, and now onto the second aim in the next slide, which is the quantification of cell death. And so here we use FlorJC or FJC to label dead and dying cells, and FJC has its strongest expression at 4 hours, but at 4 hours, the only place you're really gonna see, again, 4 hours after injury, but at 4 hours after injury, the only place you're really going to see dead and dying cells is in the dentate gyrus. So if we look back at that image in the top right, which we saw earlier, we're not looking at CA2 in red, we're looking at DG in purple. And that's what you're seeing these images down here. And so if you see in these top 2 images, again, the green cells are those dead and dying cells as a result of the injury. And really the purpose of this is to account for variability in injury, right? Because although we are trying to replicate the exact same injury every time, there is some slight variation. And so if we can create a correlation between these imaging results and the severity of the injury, it can help us to set exclusion criteria such as apnea, tail stiffening, writing reflex in selecting the mice that we will use for the behavior tasks. Because the mouse we used for the images you're seeing here were a subset of the sample. They were not the same mice we used for behavior. And so again, this is to help us set those exclusion criteria when we injure the mice for behavior tasks. And so another thing I was working on was developing a semi-automated cell counting procedure with these images. And so if you look in the bottom two images, they look a little different, and that's because they've been processed for imaging in a software called Fiji, formerly known as ImageJ. And what I've done is work on, again, creating a semi-automated protocol. So essentially, I set some parameters, such as location, because if we look in the right images, there's what appears to be a bit of an artifact in the bottom left corner, and that would confuse the software otherwise. So I can set up parameters such as location and ask Fiji to only focus on an area where I know there are going to be cells. So I would sort of crop out that area. But again, I set the parameters, and then Fiji goes in and counts everything it thinks is a cell. So if we look in the bottom left, there's all these tiny little labels about where it thinks it's seeing cells. And then I can go in again and verify whether or not each of these are actually a cell. And so yeah, that's what you're seeing in those two images. And that about wraps up everything we're doing with my second aim, which is looking at cell counts. And if we go to the next slide, we can quickly summarize everything I've done these last 8 weeks. That was, again, 1, to streamline and optimize the protocol for behavior analysis. So that's, again, what I was doing with the blood cuts. And then 2, refining a protocol for cell counting, which is that semi-automated process I was just describing. And these 2 aims relate to provide tools for future work in the lab, whether that be doing more behavior studies or taking more cell counts, or even other sorts of studies like in vivo electrophysiology. And so that wraps up what I've done these last 8 weeks. And the next slide, I'd like to thank you all for listening, and I'd like to give a special thank you to the RISE coordinators and the generous donations of Dr. John Leonard and Mrs. Elizabeth Leonard, as well as my PI, Dr. Vijay Santhakumar, and the Santhakumar Lab. If you have any questions, I'm happy to hear them.
All right, thank you very much, Ernesto. Great presentation. I think I speak for everybody in congratulating you, as well as I'm breathing for you. I was wondering, did you even breathe at all during this entire presentation? That was quite a bit of information. You
definitely, yeah, a lot of information.
You definitely demonstrated that you knew a lot about your lab and your research. And yeah, I was wondering, did you actually were you involved in lab prior to the summer?
I was actually. I began working in this lab, I want to say November, December of last year. Just before the school shut down. So I had exactly 2 days in the lab before I was no longer allowed to enter the lab.
Okay. Well, it shows you've had at least some more experience than just jumping in for the summer.
Definitely.
The floor is open to questions if anybody does have any questions they'd like to ask Ernesto directly or via any of the chat functions that we have. It is there.
I should also mention that this work has potential in other sorts of neurological disorders such as epilepsy, and that was something that drew my interest just because I have personal interest in investigating the development of epilepsy.
Oh, that's actually very interesting. Do you plan to continue this research in the coming academic year?
Yes, yes. We're still working on refining those protocols and again, repeating these trials with larger sample sizes. Yeah.
All right. Great. We have another minute. I'll just wait to see if anybody happens to have any questions. We're monitoring every which way. All right. That looks like a fun lab group.
Yeah, definitely.
Okay. Well, thank you very much, Ernesto. Great presentation.
Thank you.
We will be moving on. So thank you very much. Okay, and last but not least, we have our last presenter before the break. So we will have a 5-minute break concluding after this particular session. So Ms. Shraddha Wansley, I will give it up to you.
Okay, thank you. Good morning, everyone. I'm Shraddha Wansley. I'm a rising sophomore currently working in Dr. Sladek's lab. And for the past 8 weeks, I've been studying the regulation of COVID-19 related genes. So in the past 6 months, we are going through the global pandemic we know as COVID-19, It's caused by the SARS-CoV-2 virus, and so far we have nearly 29 million cases and 921,000 deaths. So we're still working on understanding the infection pathway, which is why my work here is important, working towards that. So SARS-CoV-2 is a part of a family of viruses with crown-like surface spike proteins, as you can see in the top graphic. This spike protein then binds to the ACE2 enzyme on the human cell surface and this allows the virus to fuse its membrane with the host cells. ACE2 is abundantly expressed in the epithelial mucosa of our lungs, as well as in the intestines and nasal and sinus tissue, which is why these are very symptomatic areas, as well as areas where we see a lot of GI problems and respiratory issues in COVID-19 patients. While ACE2 is an entry point for the virus, we can't just get rid of it because it's key to wound healing, protecting from lung injury, inflammation, and regulating blood pressure. Now moving on to the graphic on the bottom right. So that shows where once the virus spike protein binds to ACE2, BOAT1, which is an amino acid transporter, helps stabilizes this complex for a viral entry. So since ACE2 chaperones BOAT1, if any virus or drug blocks ACE2, it also blocks BOAT1 from being able to transport vital amino acid. And this can lead to amino acid deficiencies that cause painful things like colitis and other conditions. The interconnections between these two proteins, as well as how they're both independently essential to our normal body function, complicates the potential therapies and requires a better understanding of the infection pathway. The two genes I'll be studying are ACE2 and SLC6A19. SLC6A19 does code the BOAT1 transporter. Next. So now that we've explored how the virus binds and enters into the cell, these are the other questions I'm going to be answering through the course of the presentation. So first, what transcription factors regulate ACE2 and SLC6A19 gene expression? Then what tissues have the highest expression of those transcription factors? And are they the same in human and mouse? As well as is the mouse a good model for studying the expression of the proteins in the infection? Next. So to answer these questions, I'm first going to be going through this flow. So I'm first retrieving the ACE2 and SLC6A19 sequences from both human and mouse from UCSC Genome Browser. That's basically a database of vertebrate genes from which I can selectively pull out any genes of interest and the information that's correlated with it. Next. Here I entered the sequences into NCBI Nucleotide Blast. It's an alignment tool that I can use to search for sequence similarities, as well as obtain the graphic of an annotated gene, such as one that I've put on the left. Next. Finally, I make a gene map just to give you an idea of where the sequence on the gene map is pulled out from. Here's the ACE2 gene. So on the top left-hand corner, that's the ACE2 gene on the UCSC Genome Browser. And then I've zoomed in on the promoter regions. These promoter regions have then been highlighted separately on the gene map. I was only able to make this map for the human ACE2 gene since there wasn't enough information to make it for the other ones. The main feature I'm using in UCSC Browser to identify the transcription factors regulating the gene activity is called oregano. It's the yellow blocks in the top left-hand corner. You can slightly see them. They're yellow blocks. Those are the regions that transcription factors have been identified, and then I look at their roles in the infection pathway. Next. So with that graphic, as well as info from other ChIP-seq databases like FactorBook, I can use it to find consensus binding site sequences for each gene, and then use it to make that gene map on the right. And finally, I'll determine if the mouse is a good model to study the infection. So first, we're going to be looking at human ACE2. So when a patient is infected with the virus, we normally see an excessive production of pro-inflammatory cytokines, which causes a cytokine storm. STAT1 is a key activator of the type 1 and type 2 interference response, which are responsible for recruiting pro-inflammatory cytokines to the infected sites, and thus for restricting viral replication. So without STAT1, we see an increased viral replication in the lungs, the spread of the virus to the liver and spleen, and worsened inflammation. Therefore, we know STAT1 is important to the inflammatory immune response, and the only thing is that since it activates interference, we also see an upregulation of ACE2, the viral receptor. So this positive correlation pathway between ACE2 and interference may be a potential therapeutic target. The next graphic on the, on this graphic, it on this graphic, we can see how STAT3 suppresses the expression of genes that are critical to the type 1 interferon pathway. This pathway is responsible for restricting viral replication. And in the diagram, we can see that STAT3 is activated through the JAK1 pathway and then inhibits IRF7. This inhibiting IRF7 suppresses the transcription of other type 1 interferon genes, and thus it prevents them from fighting off the infection. So these next graphs, I got it from the GTEx database. It shows the gene expression of STAT1 and STAT3 separately. We can see how STAT1 is higher in the lungs and the spleen, while STAT3 is higher in the lungs and lymphatic vessels. I was mainly looking at the lungs, spleen, and small intestine quantities, since the lung and intestine are very symptomatic areas as well as main entry points for the virus. I'm looking at the spleen because it produces lymphocytes and aids in the immune response. These next three genes, also part of within the ACE2 gene body, all encode for subunits of the AP1 complex. This complex is responsible for regulating many cellular processes, including pro-inflammatory and antiviral cytokines. Therefore, viruses like COVID-19 use it to produce the massive cytokine storm we see in patients. So far, we know that the virus uses its nuclear capsid protein to activate this AP1 pathway, but in order to know how exactly it does that, we need to look at the functional domains of the virus's N protein, as well as any amino acid residues involved in AP1 activation, and these are studies that are currently being done. From here, we'll be looking at the human SLC6A19 gene, which encodes for the amino acid transporter BOAT1. So hepatocyte nuclear factor 4 is the first one that I'll be looking at. It's a nuclear receptor. It plays a role in the liver and intestines by regulating metabolism and protecting against pathogenic inflammation. So it can activate the SLC6A19 promoter. And also it is positively correlated with the viral receptor, which suggests that HNF4 alpha may play an important role in COVID-19 pathogenesis. ESR2 is an estrogen receptor. It may have a part in explaining why females have a lower disease severity and mortality than men, because testing has shown that having a higher expression of ESR1 and ESR2 results in higher immune cell enrichment, while expressing the androgen receptor results in lower immune cell enrichment in human tissue. This suggests that females have a stronger immune defense system than males, and thus better clinical outcomes. So next I'll be looking at the O minor analysis of tissue-specific expression and regulation of both ACE2 and SLC6A19 in human and mice models. The O minor tool is from the Signaling Pathways project database, and I use it to study the signaling pathways of the transcription factors I looked at earlier. Yeah, so this, I know this is a busy slide, so I'll just simplify it by first talking about ACE2 and then SLC6A19. First, I found that each receptor in humans and mouse regulates ACE2 oppositely within the two models. For example, the androgen receptor upregulates ACE2 in humans while it downregulates ACE2 in mice. However, SLC6A19 showed that the human and mice models regulated it more similarly, meaning that they may be a potentially better model to study SLC6A19 regulation in the infection. The next section is looks at different metabolic and cytokine treatment models that have been found to regulate ACE2 expression, as well as SLC6A19. We can see the main thing here is that obesity has been shown to upregulate ACE2 in humans, making it a risk factor. So overall, I was able to find that the mouse is not a good model to study the expression of COVID-19 receptors. This is because I found very little sequence similarity in regulation of the two genes, as well as little promoter alignment between human and mice. So the next thing I found was that the regulation of ACE2 by hormone receptors like androgen estrogen receptors may explain the different outcomes between male and female patients. Additionally, I also found that metabolic factors like obesity upregulate ACE2 in humans. My next steps in the project would be to look at what genetic or medical conditions play a role in the susceptibility to the virus, how the androgen estrogen receptor specifically regulate the ACE2 promoter, as well as how obesity may increase susceptibility to the virus. I'd like to thank my PI, Dr. Frances Leidick, for her guidance and encouragement, the RISE program for hosting CNAS scholars, and the generous funding of Dr. and Mrs. James Whitehead that let me carry out my research and get to enjoy this experience. Thank you.
Okay, thank you very much, Radha. That was perfect. The floor is now open. If anybody has any questions, please feel free to chat or raise your hand. And we'll give everybody a moment to type up any questions they may have. Shraddha, how are you feeling?
Pretty good. It was exciting to do all the work, and it was a good experience, I think, overall.
Great. Were you involved with this lab prior to the summer?
Yeah, I hadn't gotten too much lab time. I had started around like January, I think. So yeah, I'm planning to continue.
All right. So you actually physically got to see a lab and actually enter in.
Yeah.
Before we all got quarantined to stay home. All right, great. Do you plan to continue?
Yes, I do. I really like the lab, so definitely want to see what other work will be done in the future.
All right, sounds great. Well, looks like we're hitting our time for our first break. So we'll go ahead and start the countdown. There will be a 5-minute break. Everyone please get up and have a stretch, grab a drink, some breakfast, run to that restroom. There's a countdown already on here. And we will start right back up in 5 minutes at 10:25 with our, actually at 10:25 with our next set of oral presentations, followed by some poster presentations. All right, thank you very much, everybody. All right, I think we are officially back. All right, you can, Kendrick, you can thank Marissa for choosing the wonderful elevator music. Okay, so we are on to our next session. We already have our panelists ready. So we, without further ado, we can start with our next speaker. And this is going to be Faith or Kali Santiago under Dr. Min Su. And I think I have here. Oh, thank you very much for turning on your camera. And the floor is yours. Take it away.
All right, thank you, Nee. Good morning, RISE Symposium. I hope everyone enjoyed their 5-minute break. My name is Kalei, and for the past couple of weeks, under the mentorship of Dr. Minshu and his graduate student Priyanka Sarkar in the Department of Chemistry, research that has been conducted in the lab by Priyanka, which focuses on one cyclic peptide that was formulated by the SHU lab and our application to it on a mouse cell line. With this research, we intend to contribute to cancer drug development studies and study the transition of this in vitro application with the intention of an in vivo application onto a live mouse model. Next slide, please. So to start, metalproteinase 2, it's an enzyme that we are focusing on. It is one of a vast many of family are involved in the healing process of the body. The reason why we're focusing on MMP2 is because this has been studied for over 2 decades now due to its prominence in various diseases, but most catered toward our field of research, which is cancer. To do that, it breaks down the basal membrane of almost all cells, which can be good and bad. So because of the fermenting process, this could cause problems if there's a hyperactivity of the enzyme, thus contributing to tumor metastasis or tumors being formed. And for over 2 decades now, which poses the question of why is it still being studied for such a long time? And the reason is effects on other enzymes or other MMPs in this enzyme family. So previous clinical trials have not made through due to the fact that none of these have specifically hit MMP2. Other MMPs have been hit during these studies. Image on the screen right now. In the large box, we have the sequence of MMP2, and in the middle column, we have the percentage MMP2 holds with the rest of its MMP enzyme family. And if those other MMPs are affected, those functions impaired, which is not what we want in these studies. So what needs to be done going forward is greater drug specificity while still considering the homology MMP2 holds with the rest of MMP. Next slide, please. I've introduced the use of cyclic peptides in cancer drug development of lowering active levels of MMP2. The reason why studies have not used a cyclic peptide before, and it's different from our traditional polypeptide chain because they hold greater stability, and we can add 5 different amino acids. As you can see on the bottom left image, we have 5 different amino acids. So this is a summary of a previous publication done by the lab, and with this, we were able to see how well one particular cyclic peptide was able to inhibit cancer cell migration of a human melanoma cancer cell line. So on the top right corner, we have a cell transwell migration assay, which I will go into later on in this presentation. And on the bottom right of our cells that were successfully migrating when we implement the cyclic peptide. These are results from the previous publication cyclic peptide, and we have introduced increasing dosages of the cyclic peptide starting with micromolar. So as you can see, this inhibitor showed promising results using the human melanoma cell line intro. And with this study, we got no off-target effects on other MMPs and held low nanomolar potency. So we were able to manipulate the dosage, and it held high specificity. Next slide, please. So with this previous publication, we're branching off cyclic peptide that was successful. And our intention is to use a mouse cell line. And with this, our goal is low migration and invasion with cyclic peptide WP-HPY since it provided promising effects. And we are looking for similar results as seen on screen right now. Next slide, please. So to start off this RISE Symposium's research, we started with a blood sequence to juxtapose the human the human MMP2 sequence with the mouse MMP2 sequence. See how similar they are in structure to verify that we aren't jumping headfirst into something that's not really related to one another. And as you can see, there's a good sign with the exception of one single amino acid difference. Now, although that seems pretty minimal and we ignore it, it's not true because this is actually the region we are targeting with MMP2. So this sits in a pretty pivotal spot acid difference in the mice and human melanoma cell line, or the human MMP2 line. And our inhibitor might not trans as well as it did on the human. So we kept that in mind going forward. Next slide. The separation assay I mentioned earlier, this was one of the methods used in the previous publication done by the SHU lab. So with this test, our main intention was to simulate in vitro tumor metastasis that will naturally occur in the body. So I will walk here, starting with the leftmost panel. We have the serum-free media, which includes the mouse cells, and a membrane that's lined with a matrigel. And in the next panel, we have the cyclic peptide. So what happens here is this India that is coded in, and while doing that, they produce active levels of MMP2. And the peptide is to inhibit the MMP2 that is being produced when these cells digest the media. So they then go through an incubation period, and to do that, they migrate through the membrane to the full media. And once they're done incubating, we go through an imaging process where the cells successfully make it through the membrane, and we then measure and observe how many cells made it through. We have the results from this study, and just to compare, we have the human melanoma cell MMP2 of that. So as we can see here, we did present similar results, which is what we were looking for, which is a really good sign. So we tried to stay consistent with the usage of the MMP2 that was used. The control holds no cyclic peptide at all, so that would be normal cell migration, and with increasing levels of cells migrating. Next slide. So our overall takeaway from this study is we pose the question of, does this cyclic peptide inhibitor hold potential for in vivo testing? And a lot of discussion was done with Dr. Minshew and Priyanka to tide, so it's not perfectly, excuse me, it's ready to go for in vivo testing on a lab. We still are taking into account that single amino acid difference, and we do want to manipulate dosage of the cyclic peptide to see if we can get results closer to our inhibitory effect. So just to clarify, this cyclic peptide is not really a solution to cancer, but it is a way to alleviate and lower potential cancer growing into more severe stages. And that concludes my research. But I would do like to thank some important people who have had an important role in this entire process. That would be my faculty mentor, Dr. Minshew, for taking me onto this project, along with his graduate student, Priyanka, who has been educating me along the way, and the SHU Lab for helping me prepare for the symposium. And last but not least, the generous donations provided by Dr. and Mrs. James Whitehead. I'm open to any questions you may have at this time.
All right, thank you very much, Callie. That was amazing. All right, looks like you do have your first question from Peter, asking, what did you find most interesting from your research this past summer?
Oh, I was discussing this research and getting into this particular field of research. I didn't really know what I was going into. I was with how cyclic peptides were formed. They are a new wave of field of research, and they are being expanded on now. So I thought that was pretty interesting, and I did want to contribute to cancer studies somehow. So this was a small step, I believe, in the right direction for me, because I think one of my main questions was, is this going to be a solution to cancer? And Dr. Minshew was quick to respond, not necessarily, but it is a way to alleviate that process and prevent serious cases of cancers. And I know that with this research, I'm contributing to science in a way.
All right, awesome. Thank you very much. Well, in the interest of time, we will have to be moving along. But again, thank you again. I want to congratulate you on a great presentation.
Thank you.
All right, and we have our next presenter on deck, Mr. Leonardo Ballesteros. You can take it away.
Hi, I am Leonardo Ballesteros, and I'll be presenting on increasing conversion of xylose into ethanol through fermentation. So background, cellulosic ethanol has really great potential right now. To produce ethanol, we typically have a crop such as corn, which is sort of competing for land we can use for other things like food. So an alternative to using corn would be to use sources of cellulose, lignocellulose, and that's anything from grass to trees to anything. So here I pictured the just some grass, and you could imagine where that could, what could happen with that. Right now we have unprecedented climate change and wildfires, and we still have really high energy demands. So that grass can go to a wildfire, or it can go to potentially making ethanol and fuel. Gas isn't very clean. Ethanol is comparatively quite clean. Mostly what it releases is carbon dioxide, which is much more harmless than a lot of the emissions from gas. And it's really abundant. It's really available to find these sources of sugars, right? So in a plant, if you'd imagine, essentially this is the makeup. The blue is the hemicellulose, and the orange is the cellulose, which is a little bit different than hemicellulose. And through pretreatment, we can unlock about 90% of both of these. So now we have a lot of hemicellulose and a lot of cellulose. And in fermentation, things get a little tricky because yeast can eat glucose, which comes from cellulose, but it's not naturally able to eat xylose, which comes from hemicellulose. So in fermentation, we can pretty confidently get 90% of the cellulose to be converted into ethanol, but the xylose, it's a real struggle because that's not something that it's readily able to do. And I'll continue. So these are more specifically the sugars that we're using. C6 is easy to eat. That's like a burger. And xylose is tricky. It's a little bit smaller, and it's not naturally eaten by yeast. We like to use yeast, just to be clear, because we know a lot about yeast. It's been used for so long, and it's really understood. So what scientists have done, they've edited the genetics a little bit so that we have some proteins that are able to uptake the xylose during certain steps. So now we're able to uptake xylose, but it's very limited. Here is pictured sort of where the sources come from. We have hemicellulose, which is again in blue, and we have total sugar molecule, glucose, which is still chained up together in cellulose that we'll have to cut up into little pieces of sugar so that the yeast can eat them. Continue, please. Okay, some more background. So these are the most abundant compounds, organic compounds on Earth. Cellulose is basically half of plants, and hemicellulose is in the same order of magnitude. And here is pictured what a process would be like. Typically, the way our experiments go is we put in a certain known amount of whatever it is we're, say we're testing the pretreatment. We know how much cellulose and hemicellulose is in there. We run it and we see how much we can actually get out. And that's why we have the around 90% conversion. For the fermentation, we see how much xylose and cellulose we put in. And then through HPLC analysis, we can see how much of that was turned into, how much was consumed and how much of that was turned into ethanol. So on bioprocesses, the important part is conversion and yield. That's a quote from, sorry, below is a quote from one of the faculty at CSER. Next slide, please. Okay, so right now it's, for glucose, it's really easy for it to eat and ferment glucose, but xylose is a little bit trickier. And I propose that we make it a little bit trickier to eat glucose, but a little bit easier to eat xylose because we can do glucose so well and because we can't do xylose very well at all. And that's through adding a little bit of hydrogen peroxide. Hydrogen peroxide is a reactive oxidative species, which means that it would put oxidative stress on the yeast without keeping the fermentation, keeping it anaerobic. Next slide. So essentially, this is how the fermentation works, we put in the cellulose and we put in some enzymes, which you can see as the little red pac-man. And the yeast, it's gonna want to eat glucose first, and pretty much just glucose until it runs out of glucose, and then it'll eat a little bit of xylose. But with simultaneously fermenting and cutting up the sugar, you're going to eat xylose and eat glucose, and it's limiting what it can eat, so it will eat the xylose, and you're helping the enzymes, because the enzymes sort of get a little bit of pushback when there's too much xylose or too much glucose built up. Next slide, please. So here is really quickly this is the pentose phosphate pathway. This is where it uptakes the xylose, and it you see the xylulose phosphate. That's where the xylose comes in. It's a little bit of you use a kinase and a little modifier, a few enzymes, right, to get these molecules so that you can put them into the pentose phosphate. So this is where it enters, more or less. Next slide. Just really quickly, the glyceraldehyde 3-phosphate, that's what we're wanting to get. That's the product that we like from pentose phosphate pathway, because that can go into glycolysis and that can go into fermentation. So here, really quick, we have xylose turns into xylulose. You can read that, and it enters the pentose phosphate pathway after it reacts with a xylulose kinase, which puts a phosphate on there so that it can enter the pentose phosphate pathway. Then it goes into glycolysis, and then it's fermented into ethanol. So I think that if we put a little bit of oxidative stress, which is one of the purposes for pentose phosphate pathway, if we put oxidative stress on there, then it will want to run pentose phosphate pathway, and that will lead to more ethanol produced. Okay, so there is an issue with this, because H2O2 can be very toxic to our poor little yeast cells, especially in anaerobic conditions. When we're fermenting, it dies off really quickly. And here are just some experiments. You can build a tolerance to the hydrogen peroxide, which are the arrows, which are above, and but without it, it dies really quickly. So just in 1 millimolar, you killed about half. So that's something that we're going to have to look at. Continue. And here are the things that we are happy about. So there's an oxidative burst, which consumes a bunch of NADPH, right? Which is the thing that reduces oxidative stress. And this sort of hinders glycolysis a little bit, and it makes more of a purpose for the pentose phosphate pathway. So we slow down the one that we're good at, but we speed up the one that it's bad at. And here, really importantly, is the glyceraldehyde 3-phosphate. These are enzymes at the bottom that have been adjusted a little bit. And the glyceraldehyde 3-phosphate is much greater in abundance as opposed to without the modification, or even without the hydrogen peroxide. So here we see the WT, which is the, sorry, I'm thinking now. That's wild type, and it's not very making very much glyceraldehyde 3-phosphate. Can you continue, please? Sorry, I'm running out of time. So basically, I'm thinking we can put more stress, and we can get more xylulose out. Thank you. Really quickly, the acknowledgements. I'm very grateful to our coordinators. Thank you so much. Thank you, Dr. Richard Cardullo, for helping organize this. Thank you, Dr. Brandon Brown. Thank you, Dr. Kai, for helping me so much through this summer. And thank you, peer mentors. The research is done at CSER, our research. Thank you so much, RISE program, and thank you, CAMP again. I appreciate it.
Okay, thank you very much, Leonardo. Great presentation. Does anybody have any questions? I think we do have time for one question, or if you would like, you can type it into the Q&A chat, and Leonardo can answer that directly or type it in as well. And I did see one hand up. I'm not sure, Eric, if you wanted to ask a question or you just kind of left your hand up there. Oh, well, Dr. Katie Burnett is asking, have you taken biochemistry yet?
Yes, I have.
Oh,
that was quite difficult for me, if I'm being honest.
And since you are a camp student, were you actually involved with this research? Actually, you were involved with this research prior to the summer. When did you start?
I started last summer, actually.
Oh,
very
wow.
grateful. Yeah, I had spent a lot of time in there last summer, especially, and then not very much during the school year, and then again this year.
Okay. Do you have plans for continuing?
Of course, yeah, I really enjoy this lab.
All right, awesome, thank you. So we're gonna move on. Thank you very much for the presentation, and we're gonna move on to our next presenter, Mr. Brandon Huang. Okay, now the floor is yours.
Hi, everybody. My name is Brandon Hong, and my major is in neuroscience. And my faculty mentor is Dr. Hamshar from the Department of Mathematics. It's a shame I won't be able to see any of you beautiful people, but I'll try my best. Our research is about mathematical modeling of radiotherapy and resistance, and more specifically, modeling using ordinary differential equations. And I suppose before we go into any details of our research, there is one important question to address. Why? Why does this research matter to us? Well, let's think about it this way. There's a reason we can't repeatedly launch rockets into the sky, hoping one day it'll land on the moon. And that reason is that we simply can't afford to make these kinds of mistakes. But as our past have shown, we have launched rockets that reach the moon and beyond. Why? Well, because of mechanistic modeling and predictions made by those models. Predictions that come about from scientific inquiry. As such, our lab aims to improve physician decision-making in regard to tumor therapy using predictions founded upon mathematics, and more specifically, the predictions made regarding radiotherapy. What is radiotherapy? And what factors do we now need to consider? Well, radiation therapy, also known as radiotherapy, is a treatment that utilizes targeted radiation to kill tumor cells. But there is one caveat with the use of this treatment. Instead of only considering tumor cells as one homogeneous mass, we should now consider two types of cells that make up a tumor. Radioresistant cells, which by names are resistant to radiotherapy, and radiosensitive cells, which are by contrast vulnerable to radiotherapy. These two characteristics of a tumor are an important feature confirmed by our experimental collaborators. And because of these facts, it isn't wise to always assume the tumor is one homogeneous mass. So, well, what are the implications? Well, it would be a simple matter if we can treat each cell type in their own respective vacuums. But there is an added complexity to address. Both cell populations can interact with each other, just like interactions found in nature. And in this case, there are two specific interactions at play, whether they are competing or boosting. And here I have two figures, each using two different prostate cancer cell lines that are being experimented by Dr. Babich Cannon to represent both interactions, which are also the cell lines being represented throughout the entirety of our research. And the figure on the left represents a competitive interaction, and the right represents a boosting interaction. And looking at the competition figure, as the ratio of sensitive cells increases, the carrying capacity of the resistant population suffers. And looking at the boosting figure, as the ratio of resistant cells increases, the sensitive population's carrying capacity dramatically increases as a response. So, what exactly is the problem we're attacking here? Well, earlier we did discuss that the tumor should not always be considered homogeneous. But why? Because we would have to consider the different techniques to collect the data for either homogeneous or heterogeneous tumors. One requires an imaging scan, and the other requires a biopsy, then afterwards a cell staining. The latter being much more expensive and invasive, while the former being much more preferred because it is less expensive and less invasive. So let's look at the nitty-gritty of the math here. these are some different ordinary differential models at play here. And the one compartment model assumes that the tumor is homogeneous, which usually requires data derived from radio imaging. And I say usually because it can be fitted with data from the two compartment model. The one compartment model is also more preferred from the two due to radio imaging being less expensive and invasive. So the model in question shown below is a logistic population growth model, which we can see in the human population or even a bacteria population. And by contrast, we have the two compartment, which assumes that the tumor is heterogeneous and that it requires data derived from biopsies and is less preferred due to being more expensive and invasive. So the two compartment model represents two different equations, but the most important parameter here is only lambda. Not only lambda, but mainly lambda, because lambda represents the magnitude of interaction. A positive value would be negative in this equation, which is a competing interaction, and a negative value would be a positive, a boosting interaction. So in a perfect world, we could track the growth of tumor and radiotherapy as well, which in this case, radio treatment is shown by each sudden drop. This would require a biopsy every day, which isn't feasible due to, again, being expensive and invasive. So what can we do as an alternative? Well, hey, we can try fitting data from radio imaging. So using two compartment models with data derived from radio imaging, we can test the predictions of the one compartment model, which is the black lines of each figure. Each dot represents a scan, which gives us the total volume of the tumor. And each progressive scan throughout the day will further improve the one compartment model's prediction. Here are the figures of different ratios of sensitive cells. So the one compartment model is able to exactly predict the tumor growth with 0% sensitive cells. Well, because the tumor is homogeneous and the one compartment model is enough to have better predictions. Looking at the 50% figure, it finds it more difficult to predict the tumor growth, and especially early on with a small number of scans, since we're not tracking the heterogeneous ratio. However, if we do a biopsy each time and use the two compartment model, we could be capable of making more accurate predictions. And surprisingly the most, the 10% and 90% cells find just as difficult, regardless of whether they are closer to an homogeneous ratio, especially when sensitive cells are 90% and the prediction early on is a lot worse. So our first assumption was that we would see a graph that resembles a bell curve. Higher peaks at more heterogeneous points and smaller peaks at more homogeneous points. But instead we get something more interesting. It seems the one compartment model has trouble predicting with 90% sensitive ratios when there are a small number of scans, and also when there are less than or equal to 7 scans available, the blue and the orange lines. And surprising, considering that you would consider the more homogeneous ratios to have an easier time than ratios at 20%, 70%, and 80%, which are more heterogeneous than the latter ratios. So in addition, when more scans are available, as much as more than 10 scans, 10% sensitive cells become also inaccurate compared to the 50% mix. And it's actually especially apparent when looking at the black line. So as a result, for the higher peaks, we should now consider using biopsies to help our predictions and smooth out the higher peaks. So what's ahead of us? What's next? Well, what we plan to do next is to run simulations with radiotherapy and also simulate predictions using data derived from biopsies instead of radio imaging. And ultimately, the core of our research is to apply the two compartment model and fit into a one compartment model as accurately as possible despite these costs. It's important that we fit the models using a minimal number of scans in order to circumvent these restraints. The goal in our lab is to identify the Goldilocks zone in which we can have the most accuracy for the least number of scans. And for the distant future, we hope to help physicians make more informed decisions and introduce a more quantitative approach to medicine. I would like to thank Neil Allison Campbell Endowed Research Award, supported through Miss Rochelle Campbell in honor of her husband's memory, who passed away in 2004. He was a UCR alumni, which I also plan to be, by the way, and more notably, he is the author of the internationally renowned Campbell Biology textbook. Without either, I would not be able to be here and work on his amazing research. Thank you both very much. I would also like to thank Dr. Bavitra Cannon from Karolinska Institute, our experimental collaborator, for providing her experimental data with us. And finally, thank you to the Summer Bridge to Research program for giving me an unforgettable experience despite these difficult times due to COVID. Any questions?
All right, thank you very much, Brandon. I guess we all give you a virtual round of applause. And the floor is open for questions. And I do want to commend you for, your engagement and being a good sport for taking on and the challenge of not only doing remote research, but in a field that I know you're not completely familiar with, being a neuroscience major and working under a mathematics lab. You did a great job, really great explaining the research that you've done and how it applies to biological systems and for everybody else here. So again, thank you very much. So if anybody has any questions. Oh, I'm sorry, do you have any comments?
Oh yeah, definitely. I had no coding experience, so I had to learn coding from scratch. That was pretty stressful. But yeah, I managed to do it, and it was thank you to Dr. Hiram Cho. She's an awesome professor, phenomenal.
All right, well, glad you were able to do it in the comfort of your home in terms of these challenges of learning a new skill and new experiences. All right, and if anybody has any questions, you're welcome to either place it in the chat. It looks like, you're just having comments and about having a great presentation. Okay. And what is, I guess I'll ask you, what was one of your biggest challenges, obviously, with learning mathematical models and coding for the first time this summer?
I think the most difficult part was learning it by myself. A lot of my time in school, it's a lot more easier to learn it from people who personally are very experienced, and I can sit next to them side by side and be able to go and work through these kind of problems and these difficult portions of coding. But it somehow worked out in the end of the day, and I actually got lucky enough actually being able to do this virtually, because coding can be done virtually. So hey, thank God for that. Otherwise, yeah, that was the most difficult part. And that extra slide is a little bit more information. Basically, Monday through Friday was the treatment, and we let the tumor grow for 15 days and then continued treatment for 6 weeks. And that's kind of the surviving fraction there that helped make these figures come to life.
Okay. Well, thank you very much. So this concludes our oral presentations. So we're going to move on to the next phase of presentations, which are all posters. And this year we kind of had a challenge of trying to figure out how do we host a poster presentation virtually. And so we've come up with a new method of hopefully being able to zoom in on all these posters that the students have created. And I will start off there. And it looks like we have a comment from our camp faculty director, Dr. Brandon Brown, who by trade is an epidemiologist, but was a mathematics undergraduate major. So thanks for sharing that. So we have Ms. Morgan Brinkley and Raul Reza. They are our two next presenters. I will let you guys take it away. Thank you.
Hello, I am Morgan Brinkley.
And I am Raul Reza.
And we have gotten the opportunity to do some work analyzing zooplankton data that has been collected from alpine lakes by the Niwot Ridge Long-Term Ecological Research Site. So if we can please zoom in on our background image, you'll notice that these alpine lakes are very high in elevation. So alpine ecosystems, because of this, are kind of difficult to access, and research is a little bit understudied, especially in these lakes. And these ecosystems are very sensitive to things like climate change. One previous study noted that the ice is clearing out of these lakes about 7 days earlier over the last 33 years. So what we're doing is we're taking these data sets that have been collected since the 1990s, ongoing to the present day by these researchers on the Niwot Ridge, and running them through our Studio statistical software to get an idea of how these ecosystems are responding to climate change. So if we can go ahead and zoom out, I will hand it over to Raul to kind of walk you through what we did with this data.
Excellent. And if we can zoom into the first data analysis, we began with 3 non-metric multidimensional scaling ordinations, or NMDS for short. The important thing to understand about how this represents our data is that strongly correlated variables tend to cluster closer to each other. Now let's begin with figure 1a. This is the NMDS for zooplankton diversity metrics, and we can see clearly that Green Lake 4 forms a distinct group from Green Lake 1 and Albion Lake. This implies that the zooplankton trend in Green Lake 4 do not correlate with the trends in Green Lake 1 and Albion. It is of particular note because between the lakes, it is Green Lake 4 that is at the highest elevation. Keep that in mind. Green Lake 4 is at the higher elevation and seems to have different zooplankton trends. Now in figure 1b, the water chemistry, we observed that Green Lake 1 is the one with distinct trends. And in C, we don't observe the lakes split into any distinct grouping. C is the water quality. This implies that while Green Lake 1 has distinct chemical trends, we don't see the same kind of splitting we observed in zooplankton traits. So we assume that while some factors in the water composition may still vary with elevation, we don't expect to see the whole trend to be explained by composition factors. So to further our investigation, we decided to look at some climate variables of the region. A variable that we found of particular interest was the ice out date. Now in figure 2, right below that, we see the ice clear out date over the years for each of the lakes of interest. The ice clear out date represents the last day that the ice is present on the lakes and is related to like weather and air temperature and factors like those. What we see is that while the lake seems to trend towards a sooner ice out date, only Green Lake 4 is significant. This concurs with the findings of other studies indicating that lakes at higher elevations will experience climate change at sooner rates than their counterparts. And if we could zoom out and then zoom in on figure 3. So to continue our investigation of the effects of climate variables on our zooplankton, we compared several metrics of the diversity data to the ice out dates. This includes the smallest beta diversity seen per year to the ice out date, which is negatively correlated with that. The coefficient variable, that's standard deviation of density per year to the ice out date, see a negative correlation here again, and the day of maximum species richness to the ice out date. Here we see a positive correlation. But more study will be needed to understand this better, and Morgan will tell you a little bit about that.
So we can go ahead and zoom out here. Like we mentioned, more study is needed, but everything here is kind of backing up that there is some sort of implication that climate change is affecting these alpine lake ecosystems. So moving forward, we would like to look at individual trends of each of the specific lakes that include the temperature data as well, and compare that to the zooplankton data, but add in the data set that has the traits such as size, so that we can see if there's anything else going on that our initial search has not given us. And with that, we would like to thank our RISE coordinators, the funding from the NSF, and of course, Dr. Anderson and everyone in the Anderson lab. And we would like to open up to a couple of questions.
All right, thank you very much. Great job for being our first poster as well. And glad to see everything did work out perfectly. All right, thank you.
We're happy to be your guinea pigs.
Yeah. So the floor is open. If anybody does have any questions, feel free to type it into the chat. You can also raise your hands. Well, not at home, we can't see it. Raise your virtual hand, and we can allow you to ask the question live. Dr. Anderson has chimed in, and he's given you his seal of approval and says, great job.
Yeah.
Okay, so, and looks like, oh, Marissa's asking, where are the lakes?
So specifically, they are in the Green Lake Valley, which is on the Niwot Ridge in Colorado.
Oh, great. Do you know how this data was actually collected?
So they took a kind of net sampling at periodic dates. So they sent a little flotation device out into the middle of the lake and just kind of dropped a net in and scooped it up, for lack of a better explanation.
All right. It looks like you do have a question from Fatima. What main websites did you use to find past published studies, or was this information provided by your lab?
So the Niwot Ridge Long-Term Ecological Research Site website has all the links to their data sets and all of the studies that have used them in the past. And if you look through them, you'll notice most of the data sets that we used, at least, haven't been used in any published work yet.
All right. And it looks like you have a comment, Morgan, from a Mr. Chuck Brinkley.
Thanks,
Dad.
Dad.
Your dad? Yep.
Yeah.
Well, you have his seal of approval too. So that is also good job. Thank you very much for the two of you for being our first posters. Great job again. And yeah, we will move on to our next one. So congratulations.
Thank you.
Thank you.
Okay, so we have our next presenter is Ms. Jasmine Robledo. And Jasmine, if you are available, you can start your video. All right, thank you very much. I'm going to hide in the background here and the floor is yours.
Under Dr. Wei-Tao Chen. And today I will be discussing the comparison of forces representing pairwise interactions in center-based models. And so what are center-based models and what are they used for? In developmental biology, which studies the process of growth and change in a multicellular system, mathematical models are created. These models are capable of simulating specific multicellular events. And examples of these computational simulations can be seen in cancer and tumor modeling. Within the computational study of the multicellular systems, there exists a variety of modeling implementations. Here, we focus on the center-based model, and there are multiple simulation frameworks that implement center-based models. A potential problem can arise with this, specifically because of the accessibility of the frameworks. The center-based models rely on different default force functions where the interchangeability of these force functions is not very straightforward. This can affect how the biological behavior is interpreted and the overall quality of this simulation. So a way to help tackle this is to compare the popular force functions used in these frameworks and see if these agree. We will be looking at a specific study that compares 3 of these force functions, and this can be a quick way to assist a modeler with that selection. So if we look at figure 1, in the center-based model, cells are assumed spherical and are modeled by their centers in continuous space. Here they are represented as overlapping spheres. So by taking a closer look, the first sphere you see on the right is the mother cell, and in the middle we have the daughter cells, and these are the overlapping spheres with the cell center separation distance of R0. Then furthest to the right, we have 2 cells at equilibrium with separation distance S, and this is also known as the rest length. And at this point, the cells exert no forces on each other. In this model, the cell midpoints are tracked over time as cells interact mechanically according to the pairwise spring-like forces. Now, going back to the full view of the poster, we can take a brief look at the equation that's used to update the cell position in the simulation, which would be figure 2. And in this update equation, there are different numerical methods that can be used to solve it. So we'll just go over these real brief. There's the forward Euler method, the midpoint rule, and Adams-Bashford method. And the choice can depend on the modeler and their preference for accuracy, the amount of steps, and stability that they're looking for. So now let's break down the update equation. Starting from the left, we have the velocity of cell I equals 1 over eta. This represents the ratio of inertial forces to viscous forces. Then we have the sum of the pairwise forces acting on cell I, where I cannot equal J because cell I cannot exert a force on itself. As touched on earlier, the cells interact mechanically according to pairwise forces. In other words, the cell-to-cell interactions are encoded by force functions. These force functions, in the simplest case, can involve repulsive forces and cell-to-cell adhesion. The repulsive interactions take place when the cells are overlapping or the cell separation is less than the rest length distance. Another way to say this is the force intensity is less than 0 when separation length is less than the rest length. Then the adhesive interactions occur when cells are not or no longer overlapping but close to each other, meaning the force intensity is greater than or equal to 0 when the separation distance between cell centers is greater than the rest length. Now, when cell separation distances reach larger than rest length S, they cannot exceed R sub A, or the max adhesive interaction distance, meaning cells don't interact mechanically with one another, and the force intensity is 0 when separation distance exceeds max adhesive interaction distance. So beside the adhesion and repulsion parameters, we also have mu. This is the only free parameter, and it actually denotes the spring stiffness, and it has to be independent of whether the cells are overlapping or not. So now the force functions will produce a force intensity, which you can see in that figure, the force intensity graph. This is only a scalar force, so the direction of the resulting force from the update equation acts only in the direction of the vector between the cell centers, whose magnitude depends on the distance between those cell centers. And in figure 3, we show the relationship between intensities of the different force functions as a function of cell-to-cell separation. And starting from the top, we have the cubic force, in the middle the piecewise force, and then at the bottom the generalized linear spring force. Each of the 3 force functions are compared at 3 different stiffness values. And all 3 show that increasing the spring stiffness parameters result in stronger force intensities. This also increased the adhesion and repulsion interactions. Now, if we look at figure 4, after taking into account the results from figure 3, to test the interchangeability, of the 3 force functions, specific stiffness values along with the remaining parameters were chosen so that the relaxation time after cell separation was in agreement for all 3 force functions. The cubic and piecewise force agree well when cell separation is less than rest length, but the generalized linear spring force does not agree well for prior to cell separation, reaching 0.9 cell diameters. And then also when separation distance exceeds the rest length. This means that the 3 forces do not agree for both adhesion and repulsion, clearly showing limitations for that interchangeability. So what does all of this tell us? In summary, even using a simple center-based model like the one discussed here, there are still limitations on interchangeability of the default force functions for simulation frameworks that implement cell-based models. However, comparing these force functions is still justifiable considering the computational costs of other more expensive force descriptions, which directly affects the modeler's implementation and framework selection. And then it is to our understanding that a comparison for commonly used force functions for center-based models has not been published yet, which leads to the next steps. Possible next steps here would be to apply similar parameters to alternative force functions like the Morse potential or the Leonard-Jones potential, and then use those results to compare to the generalized linear spring force, cubic, and piecewise polynomial. Then it could be beneficial to apply to a monolayer population and see if it affects proliferation. And the end goal being to better guide the modeler to make the best suited decision according to the needs. So lastly, I would like to thank my faculty mentor, Dr. Wei-Tao Chen, for her patience and guidance. I would also like to acknowledge Dr. and Mrs. James Whitehead for funding my RISE participation. Finally, the CNAS STEM 2020 Summer Bridge to Research Program, the incredibly helpful peer mentors, and the University of California, Riverside. Thank you for this opportunity to grow and learn.
All right, thank you very much, Jasmine. That was a great presentation. looks like we're actually out of time, unfortunately, for any additional questions. But what we will do is we can keep Jasmine on the panelist side. And if you do have any questions directly for Jasmine, you can type them into the Q&A function. And she can answer them directly and publicly so everyone can see. So sorry, I apologize for that. We will move on again. But thank you very much. That was great. So we'll move on to our next two presenters. It's going to be our group before our next 5-minute break, Mr. William Troxell as well as Mr. Robert Cepeda. And both of you guys turn on your cameras. Thank you very much. And your poster is loaded, so please take it away.
Hello everyone, I am William Troxell and I am co-presenting with Robert Cepeda. We are presenting our research project for the Chaya and Cheng lab group. Our topic focuses on two cancer drugs, imatinib and vemurafenib, how they interact with multiple proteins from different kinase families, and the potential implications of the findings. For some biochemistry background, kinases are enzymes that reduce the activation energy needed to transfer phosphate groups from nucleoside phosphate donors for biochemical reactions. Most drugs are designed to target one kinase, but how may we explain the binding interactions between specific drugs and multiple proteins at the atomic level? There is a growing interest to study the downstream effects of non-targeted interactions as we realize certain substrates interact with multiple proteins because of conserved ATP binding pockets. For some drug background, imatinib is a tyrosine kinase inhibitor designed to target the ABL1 kinase to treat chronic myeloid leukemia. Its medical use has been expanded to treat other conditions like gastrointestinal stromal tumors after researchers realized it inhibits other tyrosine kinases. Vemurafenib is a tyrosine kinase-like inhibitor that targets the BRAF V600E kinase mutation to treat melanoma. Prior research shows these drugs have favorable binding potentials with some kinases from other families, but the interaction mechanisms are unknown.
Now let's take a look at our methodology section. We started gaining a general background on drug kinase interaction and whether the drug binds or inhibits to the kinase. We had a set of 52 kinases and narrowed it down based on the other lab's past research. We did this by analyzing over 90 research papers and collecting data on which kinases tended to interact with the drugs most frequently. Afterward, our PI, Dr. Chang, provided us with 10 kinases from our original 52, including 5 with crystal structures and 5 without known crystal structures. These kinases were from the tyrosine kinase TK, tyrosine kinase-like TKL, and CMGC kinase groups. The TK group contained kinases that have tyrosine residues that are a part of the ATP binding site. The TKL group is similar to TK, but they lack the same tyrosine residues. The CMGC group is a broad family of kinases that play crucial roles in the cell cycle and is named after their initials. The next task was to find reference proteins and imatinib-bound complex structures on RCSB, a protein data bank website. We decided with the help of our PI to begin our research with imatinib. We examined 4 imatinib-bound complex structures, 1 CMGC kinase, and 3 tyrosine kinases that had imatinib bound to it. We conducted a multi-sequence analysis to superimpose the kinase and complex using the Visual Molecular Dynamics program. This allowed us to compare similarities and differences between the reference structure and the complex, much like the challenge of spotting the differences between 2 photos.
Let's take a look at the table. In this data sheet, we see our 4 kinases with their PDB IDs. This list includes reference structures, the imatinib-bound complex, and other substrate-bound complexes. We were unable to find the kinase domain for ABL1, so we used an ABL1 PDB file with an imatinib-resistant mutation for our reference structure. We include data from a previous lab's work on the kinase imatinib binding affinity. Blue represents strong affinity and gray represents moderate affinity. And not applicable shows that SRC was not a part of these initial affinity trials. We include the missing residues on the far right.
Now, if we look at the figures below, here we will see some VMD screenshots with the kinase SRC. The blue represents the free-form SRC, whereas the other 4 colors represent SRC bound to imatinib. The unbound kinase has its conformation loop, the blue loop in the green circle, far away from the imatinib-bound conformation. The orange, gray, and red loops follow similar orientations because the imatinib is positioned similarly. The yellow loop is an outlier due to the different imatinib position, indicative of how drug kinase binding can vary in experimental settings. Now, if we take a look at our conclusion, our visualization trials show the kinase structures are similar when bound to other drugs. We see the differences when we examine how the binding loop changes in the presence of imatinib. We postulate that this will also be the case for vemurafenib. This research will help direct other chemists in experimental direction once we determine the theoreticals. This knowledge will be useful when developing new drugs or repurposing existing drugs for this set of kinases.
This is what we have accomplished so far, but we have future plans for the project. We will conduct a structural analysis of the remaining kinases for imatinib and vemurafenib. We will select one structure and remove the drug to prepare it for docking. Once we do that, we can take a separate drug and kinase structure and dock them using AutoDock 4 to study the positions at ideal energy levels. AutoDock allows us to run multiple docking simulations, and we can study the qualitative results in VMD to collect a general idea of where and how the ligand binds to the kinase.
We would like to thank our principal investigator, Dr. Chia-En Chang, for providing us an opportunity to apply our academic skills in a practical research setting. We would also like to thank 3 graduate students, Jian Soon, Yuliana Boskin, and Kevin Calhoun for providing direction, aid, and feedback every step of the way.
We give appreciation to the RISE program for hosting the CNAS Scholars and Camp programs. Major funding is provided by the National Science Foundation and the National Institute of Health. I would like to thank donors Mr. and Mrs. Giles Goodwin for providing generous donations to the CNAS RISE program and helping to fund this research effort. Thank you very much for listening to the presentation. Please let us know if you have any questions.
All right, thank you very much, guys. You guys did an amazing job. I'd like to open up the floor. Does anybody happen to have any questions for Robert or William? This is also the beginning of our break. Okay. Yes, we have our music up and running. Well, again, you guys are welcome, or everyone is welcome to write in any additional questions, or you could use the Q&A function, and that's something that they could potentially answer a little bit later. Otherwise, we will be going on our second and final break before our last set of poster sessions here. Okay, well, we're probably gonna need just another minute to get situated here. So in the meanwhile, I'll be moving over our next presenters into our panelist section. Okay, so it looks like we have our first presenter after the break, Ms. Judy Blanco. Judy, if you don't mind, can you please start your video for us? Thank you. All right, so we will go ahead and get started with our next round of poster presentations. Marissa is back, so you can take it away.
Hi everyone, my name is Judy Blanco. I did research this summer under Dr. Matthew Conley, and my research was in inorganic chemistry. So I'm going to talk to you a little bit about phosphorus chemical tensor analysis, and I looked at regular and strained tricoordinate phosphines. Before we begin, I am a little technologically in advance, so I don't think my Zoom feature works, but I'm going to explain everything as best as I can. So before we begin looking at regular and strained phosphines, we need a little background on what chemical shielding tensors actually are. So when we run NMRs, they actually measure our atomic environments, and the atomic environments are anisotropic. So this means that the electronic environment will produce three-dimensional tensors, and the chemical shielding tensors actually describe our relative change in the local magnetic fields relative to our induced magnetic fields. And this can produce shielding or deshielding. So when we measure this, it actually differs directionally, as you can see by the A11, A22, and A33 axes. So we actually take in these tensors and diagonalize them, and that's a figure on the right with the zeros in A11, A22, and A33. Once we diagonalize these tensors, we yield principal axes, X, Y, and Z. These axes actually correlate to molecular structure and bond directions of molecules. So our diagonalized tensors will tell us a lot about the averages of a single value that we'll observe, which is called the isotropic chemical shift. And tensor analysis is really important for my research because it provides me with insight on molecular structure and bonding distances. When we apply it to phosphine analysis, which is what I look at, it actually has applications in many different branches, including pharmaceuticals. So when we do reactions, we have products that are stereoselective, and we produce enantiomers, which are molecules that are mirror images of one another, like your hands are mirror images of one another. And so phosphine catalysts can actually catalyze stereoselective reactions. And so this can reduce waste because we're not producing an unwanted enantiomer, we're just isolating the product that we need. So I'm gonna move over and talk a little bit about chemical shift. So our chemical shielding tensor and our chemical shift tensors are actually used to calculate our observable chemical shifts. And our chemical shifts are super important because when we run NMRs and obtain these chemical shifts, they tell us a lot about the electron structure because of what I told you earlier about shielding and deshielding. The electronic structure and the molecular structure of a compound are very important because they tell us how the compound will behave when bonding and the expected behavior of the molecule based on their shifts. So we can apply this again to trigonal and non-trigonal phosphines. I want to tell you a little bit about what those are. So a trigonal phosphine adopts a pyramidal geometry, and like it says, the bond angles are 93.5. Some examples of trigonal phosphines that I looked at were PH3, PCl3, and PME3. Some non-trigonal phosphines they don't adopt a pyramidal geometry, and they're usually strained compounds. But something interesting is that their chemical shifts are usually very similar to one another, and we wanted to look at why that is. So next I'll tell you a little bit about my methods for looking at their chemical shifts. So I used Gaussian software to study the trigonal and non-trigonal phosphines. We optimized them and performed DFT calculations. And the functional and basis sets I used were B3LYP631G. And I use this functional and basis set because in computational chemistry, the basis sets used are really important because they're optimal for certain compounds, and this is the optimal basis set for a phosphine. So once I optimized the compounds, I calculated bond lengths and then compared them to experimental values. I also calculated the shielding tensors and obtained from the NMR to calculate our chemical shifts, and then visualize them so I can see how they relate in comparison to the molecules I was actually looking at. So we can take a look at my data now. And table 1 shows the isotropic chemical shifts that I obtained and calculated using Gaussian software compared to the isotropic chemical shifts experimentally determined. And so as you can see from my data, they're very, very close, and my bond lengths show the same thing. They're 0.01 amperes apart, so pretty close to each other. And what this tells me is that the functional and basis sets that I used were very accurate in producing results close to experimental values. And then we can take a look at my chemical shift tensors as well. And these are actually what we would expect in literature. So each directional component aligns pretty well with what I would expect based on bonding and distance, bonding patterns and structure of PH3 and PME3. So next we'll move to my conclusions and discussion. I'll tell you a little bit about why this is actually important. So in my next steps, I want to look at more potential strained phosphines in particular, such as TBD phospholigands, P-nicotinogen compounds, and phosphopentanes. And so strained phosphines, there's actually not a lot of NMR data on some of them. And so we can look at compounds that haven't been looked at as much and use the functional and basis sets that I've used for known compounds, because the known compounds gave us results very close to what's expected. So if I apply them to phosphines where we don't have as much, don't know as much about them, then we can expect data very close to what we'd expect experimentally, but through running our DFT calculations. So that's where I plan to go next with my research, is continuing to run optimizations and obtain data on different phosphines that there's not as much known data for. So So I want to do my acknowledgements. I want to thank Professor Conley for giving me the opportunity to do research in his lab, my graduate student that helped me so much win, the National Science Foundation, RISE for giving us this opportunity, the University of California, Riverside, camp, and my peer mentor, Karina, Nee, and Marissa as well. I just want to thank you guys for giving us this opportunity. Yeah. Oh, and any questions you guys can type in the chat or ask me. Thanks.
All right, thank you very much. Great presentation. And yeah, your Zoom functions and your posters went extremely well. Looks like you did happen to have one question by William. I'm gonna leave that into the Q&A for you, Judy, and you can actually answer that via typing. So we're actually gonna move on to our next poster presenter, Mr. Ryan Gates. Thank you for turning on your camera. Your poster is ready, so feel free to take it away.
All right, great. Thanks, Nee. So my name is Ryan Gates, and I work with the Martinez Lab in the Department of Biochemistry here at UCR. So the goal of this presentation and project was to identify YETS2 regulated genes. And YETS2 is a subunit of the attack complex, which is very important for gene regulation in human cells. So if you would mind clicking on the background images, please. So in figure 1A, we're able to see the attack complex schematic here. So here at the bottom, you can see the YETS2 protein, which actually acts as a reader protein, which reads the K27 lysine residue of H3 for acetylation or chronylation. And after this occurs, the ZZZ3 domain also acts as a reader protein by interacting with A1 at the end terminus of H3. And this reading process allows for a GCN5 protein to acetylate K4 and K9 lysine residues. So in figure 1B, we're able to see the schematic of the YETS2 protein. And you can see between the 200 and 325 amino acid sequences, you're able to see the YETS2, the YETS domain. This serves as the reading domain for the YETS2 protein. All right, would you mind zooming out, please? Okay, and so the central question we are trying to address is what are the genetic programs regulated by YETS2 in the HEK293 human cancer cell line? So if you would mind going to the methods images. Thank you. So in figure 2A, we're able to see that we started with the HEK293 cell line, and then we transfected small interfering RNA, either non-specific control siRNA or YETS2 specific knockdown RNA, in order to serve as the control and the knockdown YETS2 sample groups. After this, we lysed the cells and purified the RNA 72 hours after transfection. We then sent our samples over to the UCR Genomics Core to create the cDNA index libraries and for next generation sequencing. So in figure 2B, this is the bioinformatics pipeline that I used. It's a protocol that I got from the Galaxy forums to process the raw read sequence data into a data set that I can input into the GSEA database through the Broad Institute. All right, would you mind zooming out, please? So, and then would you mind zooming into the results and data? Thank you. So for the result, for figure 3, 3A, we're able to see the enrichment plot of genes that are bona fide by published results to be important to cancer pathways. So we're actually able to see here that in the knockdown sample group, there is significant core enrichment compared to the control sample group. And then one of these genes that are important for cancer proliferation is actually called the MEEK gene, which in 70% of cancers is actually upregulated. So here we're able to see in the knockdown sample group in the heat map in figure 3B that in the knockdown sample group, there's actually significant upregulation of the MEEK gene when we knock down the YETS2 protein. All right, zoom out, please. So, and then zoom in, thank you. And then so in figure 4A, this is the enrichment plot of meek targets, which again was another gene set library that we used. And we can actually see significant enrichment in the control sample groups compared to the knockdown sample groups. But interestingly, you can see from the figure 4B heat map that the knockdown sample groups actually have significant downregulations of these key ribosomal proteins. And would you mind zooming out, please? So this data suggests that there's a type of negative feedback loop between meek and the ribosomal proteins. And this negative feedback loop is expected to be bypassed in cancer cells where meek is amplified or translocated. So this we're able to draw conclusions from this that perhaps the YETS2 and ATTACK complex possibly act as a coactivator for meek and could provide possible new targets for treatment of meek and YETS2 dependent cancers. And this will be addressed with our collaborators at the City of Hope Cancer Center. And so our next steps will be to rerun this RNA-seq analysis pipeline without the cut-adapt program, which is pictured in the methods section. And then we will also try to apply the same pipeline that we made to a similar YETS2 knockdown experiment in non-small cell lung cancer and compare our findings. Special thanks to Kay Jang for doing all the in-lab experiments required for this poster. Also, thank you to Dr. Ernest Martinez for all the mentorship and guidance that you've provided me. And then thank you for Dr. and Mrs. Whitehead for providing the funds that we needed to buy the additional computing storage and processing power. Okay, thank you.
Okay, thank you very much, Ryan. Great presentation. The floor is open. Does anybody happen to have any questions for Ryan? And again, you can just chat. You can write it into the chat. You can type it into the Q&A function of Zoom. Or you could raise your hand and we could hear somebody else's voice besides mine. All right. So Marissa actually would like to know, what was the model organism that you worked on?
So this is a human cancer cell line, the HEK293 cell line. So I guess humans. But we're trying to apply our findings downstream to the mouse model in order for application for drug targets and stuff like that, drug research.
Okay, great. Were you involved with this lab prior to the shutdown summer, and what are your plans for the future?
Yeah, so I was actually started at this lab back in September. And then my plans for the future, I'm planning on getting more into this bioinformatics field. I'm really interested in the data science aspect and the computer coding aspect. And I'm also like playing around with like some CRISPR models, so that way we can understand this type of process better.
All right. Well, thank you very much. Great presentation. We will go ahead and move on to our next one. So thanks again, Ryan. Congratulations.
Thank you. Yeah, thank you.
All right. Okay, so Mr. Alberto Garcia will be our very next presenter. Thank you for turning on your camera. Your poster is loaded, and I will leave it to you.
Thank you. Good morning, everyone. My name is Alberto Garcia, an incoming third-year mathematics major, working in Dr. Rachel Wu's lab. And I would like to share with you my research on an examination of implicit theories of intelligence on students' bullying experiences and school belongingness. The research question driving my project was, how do students' implicit theories of intelligence affect their experiences of bullying, belongingness, and anti-bullying beliefs? Now I'd like to zoom in on the introduction. There are two types of mindset being looked at in this project that come from Carol Dweck's implicit theories of intelligence. A growth mindset, which refers to the belief that intelligence can be improved through practice and effort. Also, a fixed mindset, which refers to the belief that intelligence is innate and cannot be changed. Students with a growth mindset have shown to have an improved outlook on their ability to face challenges and improve their academic achievement. The data used in this project came from the Program for International Students Assessment, or the PISA data for short. The PISA is an assessment administered to 15-year-old students globally to determine whether schools are becoming effective at preparing students for further study or work. The 2018 PISA data included a measure of mindset and many important associations between mindset and students' academic outcomes. However, the relationship between mindset and students' socio-emotional experiences at school is less studied. So in this project, we looked more closely at the relationship between mindset and students' socio-emotional experiences at school. Now I'd like to zoom in on the methods. The participants were 4,663 15-year-old students in the USA who were given a questionnaire on their experiences at school for the 2018 PISA database. My variables included one on fixed mindset, containing your intelligence is something about you that you can't change very much. School belongingness, containing one of 6 items. I feel awkward and out of place in my school. Experience of being bullied, containing one of 6 items. During the past 12 months, how often did other students make fun of you? And anti-bullying attitude, containing one of 5 items. Is it wrong to join in bullying? The 2018 PISA database was imported into SPSS, and variables were created to run a correlation and series of regression analysis. Now I'd like to zoom in on the results. A series of regression analysis was conducted in order to examine the effort of fixed mindset on bullying experiences and related attitudes. For each model, students' gender, socioeconomic status, and academic competence were included as covariates. These factors were included as covariates to ensure fixed mindset was having an independent effect on feelings of bullying. Looking at Table 1, we see the correlation tables for fixed mindset and feelings of bullying, which reveals if there is any statistical significance between the variables. Looking to the right on the graph comparing mindset and student experience of bullying, we see the direct correlation of having a fixed mindset and experiences of being bullied. Our regression analysis tells us how likely having a fixed mindset will be able to predict reports of being bullied, a sense of belongingness, and having an anti-bullying attitude. Performing a regression analysis revealed significant interactions between students having a fixed mindset and feelings of bullying. A fixed mindset predicted an increased reports of being bullied with a beta value of 0.070 and a significance level of 0.001. A decrease in a sense of belongingness with a beta value of negative 0.066 at a significance level of 0.001, and anti-bullying attitude with a beta value of negative 0.048 at a significance level of 0.001. Now I'd like to zoom in on the conclusion. Our findings indicated a significant relationship between fixed mindset and experiences of being bullied. Also, students with a fixed mindset are prone to being bullied, feel like they don't belong at school, and are more in favor of bullying other students, although they are more prone to being bullied, which I found to be very interesting. These findings add further support for the importance of adopting a growth mindset in the classroom to optimize students' school experience and well-being. In the future, it would be beneficial to compare data across multiple countries and include longitudinal data analysis to explore how mindset and bullying experiences influence students' academic achievement and well-being over time. Now I'd like to zoom back out and give my acknowledgements. I want to thank CNAS, Persist, and RISE faculty, donors, Dr. Sue Wessler, and Dr. Connie Nugent for making this opportunity possible. Also, I want to give a huge thank you to Dr. Rachel Wu, Pamela Scheffler, and Leah Ferguson for aiding me and giving me guidance during my research experience. Thank you.
Okay, thank you very much, Alberto. That was a great presentation and very interesting. It's great to actually hear some additional research coming out of the psychology department. It looks like you're very popular and already have a couple questions, which is perfect because you ended with some time to answer. Marissa did ask, how do you think the shift to online schooling will affect or would affect your research?
That's an interesting question. I have to look more into it because most of the things that I studied with involved in classroom.
All right. And looks like William is asking, do you have an idea of whether a fixed mindset makes one more likely to be bullied, or could bullying compound the effect of a fixed mindset?
Okay. From my data, it predicted that when you had a fixed mindset, it increased the chances of being bullied. So that's what I was able to find.
All right. Well, thank you very much. We ended perfectly on time, so great presentation. Congratulations again. And we will move on to our next presenter, Ms. Jasmine Jimenez. Jasmine, if you can turn on your video. Thank you very much. All right, your poster is up and running, so the floor is yours.
Good morning, everyone. My name is Jasmine Jimenez. I am currently a third-year biology student with aspirations of going into the medical field in the future. I'm here to present my research called Neural Circuit Imbalance in the Mus musculus model of Fragile X syndrome. For the past 8 weeks and prior to COVID-19, I've been working under the lab of Dr. Irina Ethel. Her lab focuses mainly on Fragile X syndrome. So I will continue to explain a little bit about what that is and my project's focus in the background section. So Fragile X syndrome is the most common monogenetic form of autism. Monogenetic just means that it is controlled by a singular gene. And in this case, the gene is called FMR1. So in a Fragile X syndrome patient or model, we see that FMR1 is deleted and the protein FMRP is not being expressed. My project's focus is on the leading theory that holds. And this theory is that FXS arises from an imbalance of excitation/inhibition circuitry within the auditory cortex. So some symptoms of FXS include hypersensitivity to sensory inputs, repetitive behaviors, abnormal social interaction, and intellectual disability. Now, just to go back on explaining the mechanism a little bit and about how the excitation inhibition imbalance occurs, I will explain what it's like to look at an FXS model. So in the FXS disease model, excitation synapses decrease, and this decrease causes the PV inhibitory cells to not be sufficiently excited. So these PV inhibition cells cannot do their job, leading to an overall excitation of the organism and the hypersensitive phenotype we see in FXS. Next, I will be going into my methods to further explain a little bit about my project. So the way we go about this project is first we get the mice and they undergo perfusions. Perfusions is just a technique in which we rid the bodily organs and tissues of the blood to reveal a nice clean brain, which will then undergo slicing. And these slices will then undergo immunohistochemistry techniques, which can allow us to acquire these beautiful images that I will go over a little bit more in the results section. Personally, over the last 8 weeks, I have been remotely working from my computer using the J-Image software. So I have been getting these images, as you can see here. And then you can go back to the methods section real quick. Please, can you zoom in? Thank you. So what I have been doing is focusing in on these regions of interest. I have been identifying, tracing, and analyzing them. So just to go into what our region of interest is, it is going to be the inhibitory cells I spoke about before. So on these inhibitory cells, this is where the excitation synapses occur, and that's what we will be focusing on in the results section a little bit later. So by doing this, this allowed us to see what the FMR1 gene and the re-expression of it, also the deletion of it, how it affected the model on the molecular level. Simultaneously, we did behavioral testing. Although I did not do this because I wasn't in the lab, I just thought it drew a nice parallel to see what the re-expression and deletion of the gene did to the mice on a physical level. And I will explain that a little bit later in the data analysis section. So if we can zoom out and then zoom into the models used and central question, that'd be great. Thank you. So this brings us to our central question, which is how does the deletion and rescue of FMR1 gene in excitatory neurons affect the inhibitory-excitatory balance within the mouse model of FXS? To further explain this, I will explain the models used. So for this study, we'll be focusing primarily on comparing C-off mice to C-on mice. So basically what a C-off mice is, it's a mice that exhibits the FXS phenotype, so the hypersensitivity. And this is where FMR1 gene is actually deleted in the excitatory neurons. And all these images are showing is validation that FMR1 was in fact deleted. If you look at the figure 1, the bottom hand rightmost picture, you can see that there is a lack of yellow. No yellow indicates that FMR1 was deleted. Conversely, on the C-on mice, you can see that in the bottom rightmost corner picture of figure 2, FMR1 is re-expressed, as you can see the abundance of yellow. Now we will go into the results. So for the results, what we really want to focus on here are figures 3 and 4, the two bottommost photos. This is comparing C-off results to C-on results, and these are the images that I have been working on for the past 8 weeks. So if we look at the C-off results in figure 3, we can see that the excitatory synapses on these inhibitory cells are actually significantly lower than the C-on results. The excitatory synapses are indicated by small green dots that I have circled in yellow. It is easy to see that the C-off results have significantly less than the C-on. Now we will go into the data analysis section so I can kind of explain what this means. So what I have found is that in the C-off mice in which FMR1 was deleted, excitatory synapses on these inhibitory cells decreased, causing the PV cells to be inactive and leading to an inhibition excitatory imbalance and an overall hyperactive phenotype we see in the disease model. Conversely, in the C-on mice where FMR1 was re-expressed, we see that excitatory synapses increase. Sorry, I think we're having technical difficulties. Thank you. Like I said, on the C-on mice where FMR1 gene was re-expressed, we see that the excitatory synapses on the PV inhibition cells increase, causing the PV cells to be sufficiently excited, therefore restoring the IE balance. Now, just to go back to what I was saying before about the behavioral testing, the physical data actually, interestingly, matched the cellular data. With this being said, the C-off mice exhibited more anxiety, speed, and overall excitability, while the C-on mice did not. Now, if we could zoom out into the project, I will explain my conclusion. So taken together, these results indicate that FMRP expression and FMR1 expression in the cortical excitatory neurons during early postnatal development period is necessary for normal auditory cortex development. Lastly, I would just like to thank the CNAS Persist members, faculty, as well as CNAS scholars, Dr. Sue Wessler and Dr. Connie Nugent for making this wonderful research experience possible. I would also like to thank Dr. Irina Ethel and other lab members, such as Maham Race and Samantha Sutley, for being such wonderful mentors and helping me to develop my project. Lastly, I would like to thank the NIH and the NSF for funding this research, and I am glad to take any questions you may have.
Okay, thank you very much, Jasmine. Sorry about that little mix up there. Does anybody happen to have questions? You guys can chat directly or raise your virtual hand. Or you could even type it into the Q&A section and Jasmine can type in an answer for you guys. We're at 12. Looks like it's a quiet bunch today, Jasmine. It's It's almost lunchtime, but Georgina did ask, how old are the mice?
So the mice we use are P14 to P21. That just means 14 days postnatal and 21 days postnatal. And we use these mice ages because that is when the brain is most susceptible to change. Good question.
All right, looks like you got some comments here. So we're going to go ahead and move on. Thank you very much, Jasmine. That was a great presentation. So now we have Ms. Rachel Paredes. Your poster is up and running, so please take it away.
Thank you. Hi, everyone. My name is Rachel Paredes, and I will be discussing my analysis of possible sex-dependent Claudin-3 internalization following LPS-induced inflammation in a murine model. Regarding the inflammatory immune response, which is a mechanism of the innate immune response and triggered by infection from a pathogen, previous studies show that all of these processes are actually sex-dependent. Because my experiment focuses on inflammation induced by lipopolysaccharide, which is a component of gram-negative bacteria found in its cellular wall, it was important to question whether or not sex here would play a role as well. Previous studies done by the lab show that the jejunum, which is the second section of the small intestine, responds in a sex-dependent manner when induced to inflammation by LPS. Furthermore, sex differentiation has also been found regarding changes to the intestinal epithelial barrier in response to LPS-induced inflammation. And if you see here on figure 7, the figure on the very bottom left, you can actually see how the intestinal epithelial barrier looks visually. As you can see, there are tight junction proteins that lie between the cells, and what they do is they function to maintain its structure, but also regulate its permeability. What happens when there are issues with these tight junction proteins is you get what is commonly known as a leaky gut, during which your gut microbiome or your gut bacteria can actually leak through your intestines and cause inflammation in other parts of your body. And you can exit out of that figure. The protein that I was looking at here in my presentation was Claudin-3, and you can exit out of that figure, by the way. But yeah, the protein that I was looking at here was Claudin-3, which is a tight junction protein expressed throughout the small intestine, and it has previously been found in our lab to be internalized by the epithelial cells of the intestine in response to inflammation. And what that means is that it is simply taken up back into the cell and taken out of its position between the cells. My question here is whether or not Claudin-3 internalization is sex-dependent as well as spatially regulated. The sections of the jejunum from these murinae were taken out 24 hours post LPS treatment and were stained and pictured for further analysis. My objectives for my experiment were to understand how to use sophisticated software such as the NIH ImageJ software for annotation and data collection and quantification. It was also to put this data into concise tables, and most importantly, to mitigate bias when collecting my data through using a blinded methodology. And that means that I wasn't able to know which murinae were male or female, as that could affect the way that I viewed and collected my data. The methodology was very straightforward. I simply had to load the images onto the software, ImageJ. I then had to adjust the scale according to the microscope magnification that the images were taken at, which was 60x. I then had to choose the color of the image, which here I chose magenta. I then had to adjust its brightness and contrast to make sure that the image looked well enough for me to quantify and analyze. And then I had to actually quantify my Claudin-3 vesicles. So I did this according to their area size. 0.065 micrometers squared was the minimum area that I had to use in order to know if it was a vesicle or not. 0.291 micrometers squared was the minimum area for that vesicle to be considered large. Anything between those two numbers was considered a small vesicle. I then had to save my image and compile my data into a table. And my results show how the data looks visually using the software. So on figure 1, if you press there, you can see what the jejunum looked like from these mice. So figure 2 and 3 show enlarged views of what the cells look like. So figure 2 shows a positive apical cell with the Claudin-3 vesicle circled. And it's a positive cell because I could find a vesicle in there. And it is apical, meaning that I can see all the border around the cell membrane. And it is apical, meaning that it is internal to the villi and not on the surface. Whereas for figure 3, it's a positive incomplete cell, meaning I cannot see the entire cell membrane on the border of it, but I know that it is on the surface of the villi, meaning that it is completely different spatially to the cell in figure 2. It was important for me to know, to note like the different locations for where the cells were on the jejunum so that I could see if Claudin 3 is also spatially regulated as well as internalized during LPS-induced inflammation. For figures 4 and 5, if you press just like underneath figure 1, it's kind of wonky. There you can see how it looks. So figures 4 and 5, yeah, it's a little bit wonky, but basically you can see there figures 4 and 5, you can see how it's annotated. I used the letters to denote what the cells were going to be called. So for figure 4, it's labeled cell letter I, and it also displays how I would quantify the vesicles. Vesicle 21 there, hidden behind 22, is the same vesicle that was circled in figure 2, and it had a small area. Figure 5 shows an enlarged view of the annotated positive cell displayed in figure 3, and the vesicle 6 is the same vesicle that was circled in figure 3, and it is considered a large vesicle. Now, exiting out of that figure, if we go to the 6th figure, we see how I would compile my data into a concise table. So we see that I labeled the number of vesicles by the name puncta. Puncta was just a nickname that our lab used for the Claudin 3 vesicles. We can see that there were 5 vesicles in cell letter A. The areas are listed. And I also noted where they were spatially, apical incomplete. If it was on the incomplete side, then I labeled it yes. And if it wasn't, then I labeled it no. I then noted the frequency, how many cells were internal versus external, and how many were negative. In other words, how many cells did not have any vesicles in them at all. My next steps so you can exit out of that figure, my next steps for this experiment are to unblind my results, see which murinae were male or female, and then use a new software, Prism, in order to graph and see whether or not sex actually does play a role in Claudin 3 internalization. I would like to thank the Carson lab, namely Monica J. Carson, PhD candidate Paula De Silva Frost, and the rest of the undergraduate team. And a shout out to my LC mentors, Karina Morales and Gina Zuo, and Dr. and Miss James Whitehead for their generosity in funding our program. Thank you very much.
All right, thank you very much, Rachel. That definitely deserves a virtual round of applause. If anybody would like to ask any questions, you're more than welcome. I think we have run out of time, but there is the Q&A, the question and answer function. You are welcome to type in any questions you would like, and Rachel can stick around to answer them directly. Okay, so we have our next presenter. This is going through. It's a kind of a rush here, but we only have a few more presentations. Ms. Gwyneth Wong, your poster is up and ready, so you can take it away. Thank you.
Thank you. Hi, everybody. As Mee said, my name is Gwyneth Wong, and I'm a third-year biology major. I'm currently in Dr. Nandasanthur Ray's molecular cell and systems biology lab. So my project is actually an extension of a previous graduate student's project. She had worked with the POU domain transcription factors, ACJ6 and PDM3 in Drosophila melanogaster. I work with the same fly and the same transcription factors. I just want to see how those 2 transcription factors affect my specific 4 different types of family genes. So this summer, I've been using a bioinformatic tool and some previous microarray data so that I will be able to analyze the expression and the binding sites of the 2 transcription factors on my family genes. And my family genes are as follows: the DIP and DPR families. They are vital for polymerase formation and positioning. The beat family codes for proteins that actually help axons branch out, and the side family genes help axons attract. So we look at these genes because, again, they're really important to axon guidance, and the previous project that I actually mentioned has not looked at the way that these transcription factors affect these genes. So before I go anywhere else, I want to talk about my research question. Do the transcription factors ACJ6 and PDM3 have binding regions at upstream regulatory regions of DPR, DIP, B, and side family genes in Drosophila melanogaster? If so, how do ACJ6 and PDM3 affect the regulation of these axon guidance genes? So if we go ahead and head into the intro area of my poster. Thank you. I just want to talk a little bit about the Drosophila olfactory system. So in order for neurons to communicate with their targets, also known as blomeri in the antennal lobe, they go through a process of axon guidance. So on one side, we have the neurons that are trying to find their target, and at the end of their dendrites, there's actually protein synthesis that is going on, making proteins. And then on the other side, we have axon guidance genes that actually code for the proteins that act as guidance cues. These guidance cues interact with the protein synthesis that I mentioned earlier, so that the axon would be able to attract, repel, grow, or branch out into different directions. And the rate at which these guidance cues are transcribed for are affected by transcription factors like ACJ6 and PDM3. So if we can go back to just the overview of my poster, please. I'm just gonna go ahead and talk about the methods really quickly. So I use the regulatory sequence analysis tool, RSAT, to find all the sequences of each of the family genes. Then also using RSAT, I was able to pattern match the sequences that I found with the matrices that were previously found of ACJ6 and PDM3. Then from there, I was able to find the feature maps that I can analyze for the binding sites. And then taking also previous data of the microarray expressions that were already found, I can also analyze these numbers for the expression and analysis. So if we can go ahead towards the ACJ6 binding site maps in the results section, please. Thank you. So as we can see, this is the entire upstream region, and these blue lines actually represent the binding sites that each of the genes have. And then for the microarray section, you can notice that there are positive and negative numbers. These positive numbers actually represent that when ACJ6 is binded to the promoter region of the gene, it actually helps produce more proteins, whereas the negative numbers have the opposite effect. So if we can go ahead towards the PDM3 figures, please. Thank you. So the same exact thing, except there are less binding sites, and the numbers also mean the same thing. And as you can notice, there are also some missing genes on this side as well. So if we can go ahead and head down to the Venn diagrams for my conclusion, that would be great. Thank you. And so for the conclusion, we can see that there's only one gene that is upregulated by PDM3, and then there are a couple that are downregulated. And then we can also see from the map on the very, very right that there are some genes with PDM3 and some genes that have a binding site for both the transcription factors, but most of the genes actually have a binding site for ACJ6. So we can head back to the overview of my poster as I finish up my presentation. Thank you. So just overall, looking at the maps, we can find that the expression actually has not changed with these transcription factors. And it also answers our question of, yes, that our family genes do have a binding region for ACJ6 and PDM3. So some of the next steps for this project is to continue the research and to continue to analyze the microarray data, as well as find the missing genes for the binding sites and the microarray data that I mentioned earlier, as well as finding which genes are actually co-regulated, because I'm not sure if you noticed, but for my Venn diagram, the middle part is missing. And I also would like to see which axon guidance genes are also affected by the transcription factors. So before my end, I also want to thank my PI, Dr. Ray, and his lab for taking me in the beginning of this year. And I would also like to thank Dr. Nugent and Dr. Wessler for their endless support and persist, as well as Jen, Jaren, and Gina for doing such a great job in leading persist, as well as me and Marissa. Especially thank you, Marissa, for flipping through the posters. But me and Marissa have done such a good job, basically making RISE an online program overnight. And thank you to my peers and the Persist cohort for being there for me all year long. And thank you, NSF, for funding my project. If anybody has any questions, please feel free to put in the Q&A section. If anything else, I hope you enjoyed my presentation. Thank you.
Thank you very much, Gwyneth. Great presentation and right on time. So we do have 1 minute for questions. If anybody would like to ask anything, please type in the Zoom webinar chat, or you could use the Q&A function here. It doesn't look like anybody is raising their virtual hand. You can also ask directly. Okay. And I think you were also involved. Were you involved with your research prior to the summer?
Yes, I was. If you guys weren't here earlier, I did talk a little bit about that project, but it was really cool actually because I came in and the project was funded by DEET, and we actually used CRISPR-Cas9 to mutate the genes of a fly so that they would be able to detect like the chemicals in like landmines that have been buried for a very long time, and we would be able to like use drones to control them and like see where the landmines were. But that project is no longer being funded, so I'm here with this one.
Well, another new interesting, that actually sounds like a very interesting project previously done. Well, on to a new one. Well, thank you very much, Gwyneth. We will move on to our next presentation. Okay, so Ms. Krishma Uppal is our next presenter. We have your poster up. Thank you for logging in and turning on your camera. You can take it away.
Hi everyone, I'm Krishma Uppal and I'm a biology major student transferring to UCR Riverside this fall. My research is about bioinformatics and the RNA modifications. 5-methylcytosine, which is an M5C, is one of the RNA modifications and it plays an important function in regulating genes. However, whether it exists on mRNA or whether it plays an important function in mRNA is controversial. In my research, we are mapping these M5C modifications in nuclear and mitochondrial RNAs in mouse testes. And we are comparing these profiles between the ensign 7 mutant and wild-type genomes. Ensign 7 is a potential methyltransferase which is required for making these M5Cs. We use bisulfite treatment and bioinformatics to map these M5Cs in the nuclear and mitochondrial RNAs. Let's take a look at what bisulfite treatment is. Yeah, that's the one, Marissa. Can you click there? Thank you. What bisulfite treatment is. In bisulfite treatment, the samples is first treated with sodium bisulfite. When treated with sodium bisulfite, the cytosine is first modified by sulfonation, then it is deaminated and desulfonated to become uracils. Let's take a look at the methods. The mouse testes is first treated with the bisulfite first. Marissa, can you click on the background bisulfite sequencing, please? Thank you. So the mouse test in methods, the mouse testes samples are first treated with bisulfite. Then sodium bisulfite deaminates the cytosine into uracil. We then use PostScript, which is a matching algorithm to map these samples to the reference genome. If you look at the right picture, we can see how these RNA sequences are mapped. All these cytosines, which is denoted as the capital C in the reference genome, is converted to the lower T, and the genome is mapped using the bow tie. Bow tie is a matching algorithm to match the samples sequences to the reference genome. After mapping, if thymine, which is denoted as the capital T, is seen at the lower T position, it can be said that it is derived from the genome cytosine, which is denoted as capital C, converted by bisulfite. If C is seen at that same position, that position belongs to cytosine, which is resistant to bisulfite, and it is likely to be a modified cytosine, which is an M5C. We then extract these M5C mutation sites and compare the profiles between the wild type and ensign 7 mutant in the mitochondrial and nuclear genome. Let's take a look at the results. Each blue dot represents the average M5C levels in the mitochondrial and nuclear RNAs. As a result, we can see that there was an increase in the M5C levels in the ensign 7 mutant in the nuclear mRNA and ribosomal RNA. When the mitochondrial RNAs were sequenced, there was no change in the M5C levels in the mutant and wild type. This suggests that the M5C increase in the nuclear RNA is authentic. Also, we wanted to know if ensign 7 was responsible for making these M5Cs. However, if ensign 7 was responsible for making these M5C, we should have seen lower levels of M5C in the mutant. However, since we saw higher levels in the mutant, this increase is not directly caused by the ensign 7. Let's go back to the big picture. We are making, for future research, we are making new libraries of high quality to verify these results. We are also testing these results using the cell culture-based assays and also alternatively verifying these results by using the mass spectrometry. Our project helped understand how genes are regulated by RNA modifications. Also, this research can help us develop new methods for regulating cancer or viral genes. I would like to thank Dr. Weifeng Gu for giving me the opportunity to work in his lab. I also want to thank Nitran and all the donors, especially Mr. Kenneth and Nancy Gray for making the RISE program successful. And I'll also like to thank Marissa for handling my poster really well. Thank you so much. I now open the floors if you have any questions. Thank you.
All right, thank you very much. That was a great presentation, great poster, and it looks like all the zooming accentuations worked out well. Does anybody happen to have any questions for Krishma? Please feel free to type it in, or you can raise your virtual hand. And then we can give you a minute to type up anything or any questions that you may be having. Okay, seems like a lot of different data analysis that you had to do prior in a virtual setting. What were some of your challenges for this summer? And it's exactly what it's exactly what Katie was reading my mind because certain projects are, especially if you're doing biochemistry and molecular biology, it's a lot more abstract when you're first exposed to this. So yeah, what did you find most challenging about your project?
I was using the bioinformatics program because I've never learned the programming, and this project was mostly based on the Perl script, which is a computer program. So I had to learn a little bit more about that, and it was challenging.
All right. And what did you or was there anything in particular that stood out for you or that you liked about your research this summer?
Definitely that it can go further into the research of like the COVID-19 vaccinations and everything. So it was fun.
All right. Well, thank you very much.
Thank you.
Great presentation. We will go on to our next one. Thank you. So our second to last presenter today is going to be Ms. Ginny Winters. If you could turn on your camera, that'd be great. Thank you very much. Your poster is loaded by the wonderful Ms. Marissa Carranza, who's in the back. And you can go ahead and take it away.
Hi, good afternoon, everyone. My name is Ginny Winters, and over these past 8 weeks, I have worked under Dr. Richard Reddick to observe ant activity in response to air and surface temperature. We had 2 research goals in mind. The first one was to show Pongo remexic rugosa, the harvester ant, would exhibit behaviors at a greater temperature range than Solenopsis xenoli, the fire ant, due to some physiological differences. The main one being examined here is a lower metabolic rate. Secondly, we wanted to see whether certain behaviors occurred specifically in certain temperature thresholds. So if we can, please, let's take a look at our background. So an individual's metabolic rate is influenced by their weight, size, and environmental conditions such as temperature. Specifically, rugosis was seen to have a lower than predicted metabolic rate. Now, this was to be known as an adaptation for the species. Our smaller species, Xenoli, would then be exhibited in a limited temperature range in which they could behave effectively considering respiration. Our workers' tasks would then also be selective due to their priority needing to regulate their body temperature and respiration costs. Seen below is a scale diagram for our species. In red is a penny at 19 millimeters, blue rugosa at 8 millimeters, and almost half that size in green, Xenoli at 4.5 millimeters. So how do we conduct this observation? Let's move to our methods and protocol, please. So to begin with, we went ahead and distributed cookies along a pathway and retraced our steps after about an hour. Upon finding our nest opening, as you can see on the right, we went ahead and flagged the nest, assigning it a nest number and taking down the exact GPS location. We then started our day by using a random number generator and selected our nest, began our recording, and then noted the following: the date and time, the nest number, and the species we were looking at. We then focused in on an individual worker, noting their size, the surface in which they were on, and the behavior they were doing. Finally, we used a thermocouple to measure the surface that the individual had just passed upon and took the air temperature from 1 centimeter above the ground. Each of these recordings lasted for about 5 minutes or until we captured a total of 6 working individuals. This entire process was repeated until we observed 6 nests per species in a 1-hour interval. Finally, we repeated this for about 2 weeks between the hours of 6 a.m. and 7 p.m. So what did we gather? Let's look at our data, please. Well, we found that preparation behaviors such as allogrooming and refusing was primarily seen more in lower temperatures represented by our first graph. While foraging was seen throughout the day, its highest volume was shown at higher temperatures shown by our third graph. So then we saw that during the middle of the day, only P. rugosus was shown to be active. So we conducted the chi-square test of homogeneity. From this, we wanted to see if there was a difference in proportion of individuals being active and the temperature. So the first chart represents our observed number, the actual amount of individuals we saw. If there was to be no difference, our expected chart demonstrates the numbers we would have had to see. Upon calculating, we found that our p-value was greater than 0.001. This indicated that there was a significance. From this, we did see that highlighted in yellow between our 30 and 34 temperature range, there was no Xenoli present. However, if we were to prove our null hypothesis, we would then need to see 12 individuals. So from this, we rejected the null hypothesis and concluded that rugosa was considerably more active than Xenoli. Pictured below are 4 images of some behaviors. The first one is a winged individual being guarded on her left. Second is an individual refusing or removing a pile to a discard location. A smaller one is shown in blue. The third one is a nest entrance at around 6 a.m. And then the fourth one are some individuals allogrooming, in which they face each other, take off some debris, and then also take part in some social behavior. So what can we conclude from this observation? Let's move on to our conclusion, please. Well, we found that early mornings and late evenings had a pattern of 2 activity cycles, coldest temperatures showing preparation behaviors and mid-temperatures showing more strenuous ones such as foraging. While the hottest part of the day showed no activity for either species, it did display that temperature threshold that the workers' metabolism limitation forced them to retreat back underground to avoid desiccation. We additionally saw that rugosus did benefit, in fact, from this lower metabolic rate because they had an expanded activity time frame, and within this time frame, as we saw in that second graph, they had limited competition. So some future steps we would like to take is continuing this observation in a seasonal pattern, expand our observation time between 5 a.m. and 9 p.m. This is because Xenoli represented limited data within these 8 weeks. There's perhaps a preferred temperature threshold we can capture them at. We would then want to evaluate and focus in on the individual's foraging and the amount or percentage of those guys actually bringing in resources. Finally, we would like to address the much larger question of climate change and resource availability, and whether we can expect these two species to partake in some resource partitioning. Finally, as we zoom out, I would like to give a thank you to Dr. Purcell's Six-Leg program funded by the USDA NIFA grant, the John and Elizabeth Leonard Family Foundation, the Summer Rise to Bridge Research Program, RISE, Dr. Richard Reddick, Rebecca Kine, and the University of California for making this presentation possible. Thank you. I would like to open the floor for questions.
All right. Thank you very much, Jenny. You did a great job, and especially considering there are tons of people to thank and funding sources, it can definitely be a tongue twister. I have to memorize all of them, so. The floor is open. If you guys happen to have any questions for Jenny, please feel free to type or raise your hand. But I know you in particular had a lot of, you actually had real work and occasional field work to be done even during social distancing. Can you tell us maybe a little bit more about that experience?
Yeah, so it was quite an interesting experience. We actually went on a hill behind UCR, hiked and located our nest. It is a popular mountain biking path, so we did have to wear masks, which did make it difficult in the event to keep our social distancing. But for the most part, since it was fairly secluded, it made it pretty nice to get out of the house.
Nice. It sounds like you visited, and it looked like you visited the infamous 5C Hill. It's aptly named after Biology 5C, which is the third quarter of general biology here at UCR. Looks like you do have a question from Mr. Enrique. What activities do ants perform during the nighttime?
So we were unable to capture it during the nighttime, unfortunately. That would go in our future steps, perhaps using a red lamp, so then we wouldn't have to bring any real light, so we could capture the insects doing a more behavior. But hopefully they'd be out, still foraging. We would have to see.
All right, awesome. Thank you very much.
Thank you.
All right, so your time is done. Congratulations, very great presentation. And now we have our next group. Our last presentation of the day will actually be done by a group. There will be 4 students. So I will let you guys take it away. Your poster is up and your time will start now.
Thank you, Nee. Hello, everyone. My name is Paul Suwatnodam, and our study is based on the effects of blepharisma morphology on the population dynamics under Dr. Kurt Anderson's lab. Intergroup predation, which is known as the IGP, is a normal observed ecological interaction where the IG predators both feeds on the IG prey and compete with it for a shared resource. The purpose of this research is to understand how the foraging strategies of the IG predators is mediated by changes in morphology under different conditions and how these impact population dynamics of the IG prey. We use the 3 trophic level model system, which consists of blepharisma species, our model unicellular protist that undergoes a trophic polymorphism as the IG predators, the bacteriovorous protist tetrahymena species as the IG prey, and the bacteria as a basal resource. To investigate the studies, we manipulate the initial bacteria and tetrahymena density to observe changes in blepharisma morphology. The result, which we will be discussed, will contribute to the overall understanding of how environmental context shapes complex species interaction. Next, we will go through the methods of our research experiment.
Hi, everybody. I'm going to be introducing the method section. My name is Tuet Nguyen. But before we get started with that, I'm going to cover some terminology that we'll be commonly referring to. First is a macrostome. Macrostomes are the larger form of the blepharisma protist that we are studying. And this form exists mainly to eat other larger protists such as tetrahymena and other blepharisma as well, because it's a cannibal. The other vocab term is microstome, which is a smaller morph of the blepharisma protist we are studying. This one is smaller and it is more nonspecific in what it eats, so it eats more bacteria and other floating particles. When we refer to the term trophic polymorphisms, what we mean by that is that the blepharisma protist is able to switch between these 2 morphs. Generally, it switches mainly from microstome to macrostome, and the reason why it does that is because it eats tetrahymena, which is another protist, and it's about 1/3 of the size. So when it eats, it just gradually gets bigger and it becomes a macrostome. And as you can see here on the right is the pyramid that demonstrates the 3 trophic level model system that Paul just mentioned. And towards the bottom, you can see a chart that displays the different treatments that we put the blepharisma in. We alter the levels of tetrahymena and bacteria appropriately according to the treatments. And to look at the results, we will be discussing that next.
Great, thank you, Tuet, for talking about the methods. My name is Jonathan, and I'm going to be talking about the results now. So starting off with figure 1, we constructed a box plot based on the final measured area and the different treatments, including the stock treatment. So we can see that the sizes of macrostomes in each treatment collectively trended downwards, and based on the NOVA test that we ran, which concludes that the macrostome area treatments are significantly different compared to their initial stock areas. Though the microstome treatments are not significantly different from the microstome stock initial areas, we can see somewhat of an uptrend towards going up and increasing in final size. So moving on to figure 2, which is really similar to figure 1, this is pretty much another box plot, but this time it's the difference in area for blepharisma. Majority of the microstomes with no significant difference increase, while the majority of macrostomes decrease in size, with the exception of low tetra high bacteria treatment, which is likely due to the high tetrahymena population. Figure 1 and 2 basically go hand in hand into explaining the final sizes and different trends of the blepharisma sizes in each of the treatments. A possible explanation for our results is that we handpicked the biggest cannibals, biggest cannibal microstomes from the stock models, and a change in diet from consuming smaller microstomes to tetrahymena or bacteria. It makes sense that the big cannibal macrostomes would decrease in size because it's not advantageous to actually maintain its huge size. Another thing to notice, which is pretty interesting, is that the microstomes in the the no tetra high bacteria treatment actually died unexpectedly due to unknown reasons. So now moving on to figure 3, these graphs pretty much examine the change in population size for tetrahymena, bacteria, and blepharisma. Looking at 3A, figure 3A, the low tetra high bacteria treatment, the tetrahymena with their aggressive eating behavior grew really aggressively compared to the other 2 treatments. And this is highly due to the high bacteria environment, and this treatment essentially acted as a high tetra high bacteria treatment. Looking at the changes in these population graphs, we can see the general trend of all 3 species, such as how tetrahymena generally increase, whereas the majority of bacteria decrease, as seen in figure 3B. Another cool observation that we can see is that the low tetra high bacteria treatment caused a decrease in blepharisma in both microstomes and macrostomes. And this is highly likely due to the enormously high levels of tetrahymena, resulting in high interspecific competition between the 2 for bacteria. Finally, by connecting figure 3C, figures 1 and 2, we can see that the macrostomes increase in population size while decreasing in average area. And this further suggests that the macrostomes are actually actively dividing to decrease size due to the environment of tetrahymena and bacteria not sustaining its cannibal diet. So now I'm going to pass on to Kevin to explain the conclusion as well as our further future implications.
Thank you, Jonathan, for talking about the results of our experiment. My name is Kevin Blatton, and I'll be concluding our presentation as well as talking a bit about future implications. Through our experiment, we learned a multitude of things. As Jonathan explained, we found that blepharisma macrostome sizes tended to decrease in size, and that microstome sizes increased in size depending on what treatment they were in. We also saw that macrostome densities increased at a faster rate than microstome densities, which suggests that macrostomes divided at an increased rate, which in turn would cause a decrease in their sizes in different environmental conditions. Lastly, our results suggest that the reason we saw microstome and microstome blepharisma areas converge to an average size was due to macrostome decreasing in size by dividing into microstomes, possibly because it is more energetically favorable to decrease in size rather than maintaining its large morphological size. And we saw an increase in microstome sizes, possibly due to the presence of tetrahymena, prompting them to try and increase their size so they'd be able to eat them. We saw that these morphological changes that blepharisma underwent were largely heterogeneous in nature, and we believe that this was the case in order to decrease interspecific competition by partitioning resources when food resources became scarce. The experiment that we conducted really sets the stage for future interactions or building of models that could contribute to helping future studies on food web dynamics, population densities, and improved conservation of species in general. Marissa, would you mind zooming out for me? Thank you. We would just like to give a special thanks to Dr. Kurt Anderson, Matthew Green, Clara Woody, Megan Whitesell for all of their support and knowledge provided throughout this experiment. We would also like to thank the National Science Foundation Summer Bridge to Research and Dr. and Mrs. James Whitehead for their generosity in funding our experiment. Thank you everyone for listening to our research, and we are open to any questions at this time.
All right, thank you very much. Great job, guys. And you guys came in just right on time. We do have a little bit of a buffer in terms of questions. Does anybody happen to have any questions? You can go ahead and type into the chat. There's a Q&A. There is one hand raised by Ms. Joyce Bauteng. I don't know if Kevin may or may not be related. And I don't know if she would like to ask any questions directly. Her mic is unmuted. Unless you just wanted to embarrass you, I don't know. And Dr. Anderson is giving his seal of approval. Great job to everybody. All right. So you guys are actually one of the larger groups working together on a project as well as presenting one. How is that experience like for the summer?
It's actually a pretty interesting and amazing opportunity for me to work with Dr. Anderson's graduate students and along with other undergrad students. I feel like it actually built more connections for me to be able to get to know more people. And then especially working in the group setting through like online media, I feel like we actually could connect, still be able to connect in like a interpersonal relationship wise. So yesterday, for example, yesterday we kind of just talked about like our experiences, and then later on we still continues with what we were doing for the presentation practicing. So it's actually a good experience for me.
All right. Well, Marissa did have a question for you guys. How and or where were the protists collected?
Right, so the entire experiment was done in the Anderson lab, which is in UCR, and it was done by our awesome grad students, Megan and Clara, and they both contributed in doing all of the experiments, and they just they collected the results and all the data, and they just gave that to us to analyze. So it was all done in the Anderson lab.
All right, thank you very much. Okay, well, this actually comes to a close and the conclusion of our symposium for the first day. Yes, great job to everybody. Great job to the final group. And yeah, we have 2 more full days like this. So the link that we gave you did have the presentation. Tomorrow's symposia is planned to be the exact same time. It'll run all the way till 12:45. And the very last day will be cut a lot shorter. So I know everyone must be really hungry. I know I am. Thank you again for attending and viewing everybody's presentations, asking questions. This presentation is recorded, so we will make this available for everybody once it is once we actually have the link. So again, great job, everybody. Please go grab some lunch, have a break. Thank you, Marissa, for chiming in. Marissa is our Wizard of Oz. Just so you guys know, she is on the UCR campus, logged into what is hopefully the better internet as opposed to her home office. And yeah, thanks for actually jumping in here. So we'll go ahead. Please, Marissa, find an open bathroom. I know everything's locked. You deserve a long lunch and just relax for the rest of the day. And we will see everybody tomorrow. Thank you very much.